All right, welcome everyone to our second day of our 32nd annual River Symposium, this year's theme being Changing Waters. Just a little bit of housekeeping before we get started. As many of you have seen, we've got coffee, tea, and some snacks at the front of the room here, so of course help yourself. Bathrooms are again located just up past registration. If you keep on going down that hall, you'll see the bathrooms at the end there. Lunch will be served at noon at the Propeller again today. And again, if you weren't here yesterday, we can help you find that because it's a little bit of a maze here. Booths are located out in the foyer, so please again check them out during the breaks and at lunch. We also have a map from Canadian Ocean Literacy Coalition that's upstairs in the foyer as well that you can go and check out and see all the waterways that Canada has. Today in Salon D117, we have an event called Fish Tank, so that's continuing from yesterday, and we have youth from the area participating in that in a case study challenge. So if you see them at any point today, you can ask them about the case study and see how it's going. This afternoon, we have the River Strategy Annual General Meeting that starts at 2:30 p.m. And the River Strategy, if you aren't familiar, it's a hub for anyone interested in maintaining a healthy and beautiful St. Lawrence River. Everyone is welcome to attend the meeting, learn more about the initiative, and engage in some discussions about the health of the St. Lawrence River, so we'd love to see you there. During the breaks, we will share slides from artists that have submitted that are nature-based, and a lot of those being from our eel art competition, so we hope you get to enjoy some of those. I'd also like to take a moment to recognize and thank our sponsors for this year's symposium. So we have our Pike sponsors, Ingredion, Ontario Power Generation, the St. Lawrence Seaway Management Corporation, and the Canadian Ocean Literacy Coalition, also Cornwall Tourism Development Fund. We have our Bass sponsors, Rogers Marina, Walmart, the Kawartha Credit Union, the Great Lakes Fisheries Commission, and the Bracewater Centre at McGill University, as well as the International Joint Commission and the Catholic District School Board of Eastern Ontario. For our perch sponsors, we have the St. Lawrence Parks Commission, the Outdoor Learning School and Store, Alt and Alt Law Offices, and then we have our media partner, Water Canada. The River Symposium would not be possible without their support, so thank you. Again today, we have a daily draw prize for gift cards to the Outdoor Learning Store. So you're welcome to put a ballot in. Even if you put a ballot in yesterday, you can put one in again today. Go see the registration desk and they will set you up with that. And we'll be drawing that at the end of the day before the River Strategy AGM. Okay, welcome everyone. Thank you for being here today. And now we'll get started with our plenary. So for our plenary today, we have Dr. Stephen Lougheed. He's a professor of biology and environmental studies at Queen's University. He holds a Bachelor of Science in Molecular Biology and Genetics from Guelph University, a Master's of Science in Zoology from Guelph University, and a PhD in Ecology and Evolution from Western University. He was director of the Queen's University Biological Station from 2011 to 2025, and was Bailey Chair of Conservation Biology over that same span. His research encompasses three broad themes: quantifying the interacting evolutionary, geological, and climatic forces that shape the evolutionary history and diversity of vertebrates, quantifying human impacts on reptiles and amphibians of conservation concern using genomics and geomatics, and using environmental DNA to map species distributions and monitor species diversity over time and space. Stephen has 40 years experience working on birds, amphibians, and reptiles, and other vertebrates of the Americas, particularly in Canada, Argentina, Brazil, and Mexico, with significant forays into China, East Africa, and Australia. Steve is truly a global researcher. Steve has supervised or is supervising over 50 graduate students, 10 postdoctoral fellows, and over 100 undergraduate theses or mentorship students. With his students and collaborators, he has published over 175 peer-reviewed articles, government reports, and book chapters, and has attracted over $28 million in research funding to Queen's University. He is a lifelong naturalist with a passion for birds and herpetofauna, inspired by his maternal grandmother and mother, and is a dedicated nature photographer and bibliophile. So please welcome Stephen Lougheed.
Thank you so much for that generous introduction. He sounds wonderful. So it's a great pleasure to be here. Thank you so much for inviting me to the River Symposium to speak to you today. And also thank you especially to the organizing committee. I know what a monster something like this can be to organize, so it's really a pleasure to be here. So what I would like to do today over the next 45 minutes or so is to talk to you about connections or connectivity. This is a graphic I asked my daughter to make for me for this talk, and it emphasizes connections in ecology, genetics, demography, conservation, which is sort of the focus of a lot of the work that we do in our lab. But it also emphasizes that we can speak of connections among human populations and mobility and things like that. So before I dive into 3 case studies that were done by some of the wonderful students and postdocs in our lab, I'm going to talk a little bit about human connections and human connectivity. So I think a lot of Canadians, perhaps not all, take great pleasure in knowing that Canada is among one of the most multicultural countries in the world. Just as an example, Toronto is touted often as if not the most multicultural, one of the most multicultural cities in the world. Over 55% of Torontonians identify as visible minorities, and over 200 different languages are spoken daily in Toronto. And you will know this when you go to the city of this wonderful diversity. And of course, what that means is those people from these myriad cultures represent rich diasporas from their home countries, and they've brought that culture and connectivity, and for me, importantly, cuisine and music and literature to Toronto. So that's a connection that I think is very important, and it speaks to mobility, and it speaks to sort of the unity of humans. I sound a little bit fluffy there, but I do believe that. But I'd like to make a more sort of tangible point about connections. We know over the last 2 or 3 centuries that we've seen an increase in technology that can really mobilize and move human populations. So this is a graphic that was put together by the Air Traffic Action Group for data that comes from 2023. Now, before I did research for this talk, I didn't know of that particular organization, but they track flights every year. So these are data from 2023. There are 3,200 airports represented in this graph. These are commercial airports. We're not talking about private, smaller. I'm afraid Kingston and Cornwall probably are not represented on this map. And these are 60,000 different routes that are captured here, major commercial routes. And what you can see is the dots represent the airports. The size of the dot is essentially how much traffic there is, and then the lines will connect these airports with respect to the different routes. So what this represents in 2023, and remember this was a down year, we're just recovering from the pandemic, 4.4 billion air passengers are captured here, so about half the number of the global population were in the air at one point. 1.8 billion of these were traveling internationally. This represents 35 million commercial flights and 8.17 trillion passenger kilometers. Now, Canada is a sort of minor part of this when you're talking about massive countries like India and China and air flight there. But Pearson is among the 25 busiest airports in the world. You may or may not know this. It has 31,000 connections annually, 50 million passengers, and 200 different destinations. So Canada is connected. and we're connected to all of these amazing places. I teach field courses all over the world, and it's interesting to look at Montreal or Toronto and where you can get to fairly easily, usually with a horrendous layover in Heathrow or something. Now that's human movement. We can also talk about cargo, movement of goods. And so we can look at shipping. And here this is an emphasis on trade. So this again is a representation, it's a little bit older, it's a maritime traffic density map. And these aren't the Queen Elizabeth II pleasure cruises, this is movement of cargo. And it's 12.3 billion tons of cargo that were moved in 2023. And the green lines are major sort of transit routes. So we see significant movement of goods. And just as an aside, I was listening to the CBC the other day, and they were talking about carbon footprint, and it was suggested that shipping like this represents about 3% of the global emissions. So that's a significant contribution. I think they were talking about electric ships and the new move to move away from diesel-powered ships and so on. So what does this mean? It means that we've seen massive globalization, and there are good aspects to this. The good aspects, I think, are that southern economies that were facing sort of pressing poverty issues sometimes can be lifted up economically because of connections to wealthier markets like our own. We have the ability to explore other cultures, and I think that brings us together in a time of increasing xenophobia, so I think that's a good thing. But of course, there are bad aspects to it. I've already alluded to the carbon footprint of shipping. There's certainly a carbon footprint to air travel, so we can talk about that. When we have globalization of trade, that means there are great pressures on countries like Brazil and Indonesia to produce soybeans or beef, and that results in clear cutting. We see the spread of invasive species. We see the spread of invasive pathogens, not just human pathogens, but pathogens of wildlife and plants. So that's bad grading into ugly. This is connectivity. This represents connectivity, of course. There we go, it does work. I've got an animation here. So humans, of course, did not invent mobility. And we know that there are wonderful migrations that take place every year. There's the wonderful migrations of barren land caribou that occur every year. I teach in East Africa, in Tanzania, Kenya, and Uganda. And of course, the Serengeti represents a wonderful migration of wildebeest. We know of the bogone moth in Australia that migrates altitudinally, the wonderful migratory monarchs that move southward to overwinter in the Oyamel forests of Michoacan in Mexico. But the most spectacular example of this is avian migration, bird migration. And so this is an organization called MoveBank. They're out of the Max Planck Institute of Animal Behavior, and they collate animal movement data. Now, most of the data that you're seeing here, these are for all animals in the year 2017. Most of these are birds. That's because there are a lot of bird heads out there, otherwise known as ornithologists, and there are a lot of avian researchers. And some of the most spectacular movements of animals involve birds. So you probably know we're just finishing up the migration now. I have white-throated sparrows and I hope fox sparrows in my backyard. But we've just witnessed a massive migration of birds out of the Great Boreal Forests, moving southward all the way potentially to places in northern Argentina or even all the way down to Tierra del Fuego. And this graphic that you saw up here represents all of the major flyways. So we have a flyway that links North America and South America, and in fact, multiple flyways in the Americas. We have a flyway that links Europe down into Africa. We have a Central Asian flyway, and we have a flyway that represents Eastern Asia, China, and Russia, all the way down to the Australasia and parts thereof. But to give you a number here, it's estimated that right now we're just finishing up on 3 to 5 billion birds having moved out of our boreal forests up into Alaska. And this number used to be much greater. I don't know if you've ever done this. I encourage you to do it next year. Every year in the fall, I go lie on my back on the deck or in some park, just some strange and late middle-aged man doing strange things, and Kingston is perfectly accepted. And if you lie there in late September, you can hear the birds migrating over. And they actually track them via radar, there are so many. But in my youth, I was born in 1960, so in the 60s and 70s, that number would have been double or triple that. So imagine 10 to 15 billion birds flying above your head. Not all at once, but it's a pretty amazing thing to think about. So that speaks to connectivity on a massive scale, on a continental scale, and we're going to focus in a little bit. But I did want to showcase some of my own photography a little bit, and I wanted to put an avian face to this phenomenon. I don't know which I put first. I don't know if any of you know what this wonderful bird is. I know that they occur around Cornwall. This is called a Virginia rail. And a Virginia rail is a wonderful water bird that is emblematic of a healthy marsh. And so in a healthy marsh, fully functioning, you'll find all kinds of wonderful critters that can only exist there because of the fully sort of functioning ecosystem. So many of you will not have seen a rail because they're more often heard than seen. You have to know what the little squeaks and burps are in the forest or in the marshland to know that that's a Virginia rail. And sometimes you can call them out and they'll reluctantly and furtively come out and look at you. But my point here is that they cannot overwinter. They cannot persist in the marshland in the Canadian winter, so they must move. And for them to move, they must be connected to other marshlands, all the way down to the Gulf Coast, the Gulf of Mexico, I insist, and all the way down into Mexico. And that connection is really important. So it's not just that they have to fly, they're not active flyers. That connection has to include contiguity or connectedness among different intact wetlands. This is another photograph of mine. I don't know how many of you know this wonderful bird, one of my favorite wood warblers. It is a cerulean warbler. It's a male cerulean warbler. I took this photo at the Queen's University Biological Station. This is an 8 to 9 gram bird that in 2 to 3 weeks can make its way from the breeding grounds in Canada all the way to the montane slopes of Venezuela, Colombia, down through Peru. I challenge my students sometimes. I said, I'm not giving you any food. You can have one change of clothes. You don't have to do this without clothes. But now you have 2 weeks without hitchhiking to get down to South America. Good luck. But that's what these things do. And to do so, they have to have intact forests. They have to have stopover sites. So again, that move bank figure sort of condenses an awful lot of really important connections and really important dispersal ecology that we have to understand. And we're still wrestling with how migration arose. So this is a sort of fundamental thing in science. How did they, because 20,000 years ago, this was a couple of kilometers of ice. They're recent, sort of, they recently arose here. So that's speaking about connectivity, natural connectivity, migratory connectivity on a continental scale or on the scale of entire ecoregions. But of course, connectivity is important at a landscape scale, a regional scale, or a within landscape scale. So here we're looking at Cornwall, sort of at the heart of this area. This is the Algonquin to Adirondack region, you will know, but it's also the heart of the Frontenac Arch Biosphere Reserve. And there's also a biosphere reserve to the south in New York as well. And what this represents, this area, is the most important forest connection north-south east of the Rocky Mountains. This area is that important for connections. And this connectivity, and I would say also, as we'll see in the case studies I present, contiguity of habitats is incredibly important for ensuring that species and people continue to flourish in this region in perpetuity, we hope. Perpetuity is a long time. Eventually the sun's going to explode and the earth will disappear, but that's a few billion years into the future. We're going to look a little bit closer. So that's just a little bit about connectivity, human connectedness, and it's about natural connectivity, sort of a little bit across hierarchical scales. We'll return to that theme in just a minute. But now I want to give you some fairly stark statistics. not a pessimistic guy, so this comes across as a bit pessimistic. We'll try to get into some more optimistic stuff. So this is an organization called the Intergovernmental Platform on Biodiversity and Ecosystem Services. It's associated with the United Nations, but it's not of the United Nations. It's sort of a parallel organization. And they took a report that suggested our most robust estimate of the number of species on Earth is about 8 to 9 million species. This is excluding viruses and bacteria. If you know the literature at all, you will know that only 1.5 to 2 million species have been described. And when I say described, I'm using that term loosely. What that means is they give a scientific name. It doesn't mean we understand their ecology. We know surprisingly little about a surprising amount of things. And what they did was they took this number, and through a variety of different means, they came up with an estimate that there are probably about 1 million species that are at risk of extinction in the near term. Perhaps not in my lifetime, but in the lifetime of the younger people in this room. And what that means is that probably 60 to 70% of those remain undescribed by science and will potentially disappear before we ever describe them. They might be little microlepidopterans, tiny little butterflies, or fungi, or tardigrades, or things like that. But we are still discovering new species of birds and frogs, cervids, which are deer, which is surprising. And so there still is a lot of sort of primary discovery. So 1 million species. We'll often see cited the red list. There are 20 or 30,000 species at risk, but those are species that UNEP, the United Nations Environmental Protectorate, has a program rather, has actually evaluated. That number is probably closer to the reality. You may know that the World Wildlife Fund, or Worldwide Fund for Nature, depending on where you are, every few years does an evaluation of the state of global biological diversity. And they put out a Living Planet report, which is a really amazing report, usually with different foci, and you can look at sort of different themes in biodiversity conservation and potentially biodiversity loss as well. But they also come up with this composite measure called the Living Planet Index. And we actually work with people in China on the Yangtze River to come up with a Living River Index there. And you do similar kinds of things here along the St. Lawrence, of course. But here we're tracking over 5,000 vertebrate species, if you can see the legend, and over about 35,000 populations of those species. And what they're doing is looking at whether they're increasing, stable, or declining. And they come up with this composite index. And they've been tracking it or have data from 1970 to the present day, more or less. And so 1970, I was a 10-year-old boy. And so since I was 10 years old, over 70% of biological diversity by this metric has been lost. So a pretty dire circumstance. And one of the problems is we are aware of diversity that we grew up with and unaware of the diversity that indigenous people of this region would have experienced. And that's an important thing to note, I think. The dominant drivers of all of this are very well known. So you don't really have to go to study it to get the rank order of the importance of these things. So if things are shifting a little bit. This again is from the WWF report. Habitat loss, fragmentation, and degradation are the leading cause of biodiversity loss and species endangerment. Overexploitation, particularly if you think about fisheries. Contaminants in the environment are an important thing, and certainly have been a focus of some of the wonderful work at the River Institute, sort of understanding this and remediating and restoring. Invasive species are an increasing problem, and emerging diseases. So there are a lot of emerging diseases. You can think of avian malaria, for example, West Nile virus, and things like that. Anaplasmosis, which is carried by black-legged ticks. We just looked after a dog that turns out had anaplasmosis that had picked up from the tick bite. So it's not just Lyme disease. So far, I don't sound very optimistic, I'm sorry. But all of this, of course, is underlain or exacerbated by climate change. And so what that means is we really, if we want to rectify these things, if we want to remediate, if we want to contribute to tangible action in conservation and habitat restoration and preventing further loss of biological diversity, something swinging out at me aggressively. We have to understand what is going on with different systems. And so what I'd like to do is to present to you three sort of, I call them research vignettes, because I'm not going to go into great detail. I'm not going to give you grotesque, detailed statistics or anything like that. I'm going to skim a stone across the top And hopefully we can understand these three different studies. So I'm going to talk about, I said the cerulean warbler was one of my favorite snakes, birds. I'm going to present one of my favorite snakes, the fox snake. I love snakes. I'm going to talk a little bit about some wonderful work that was done with the River Institute, actually led in many ways by the River Institute, Amphibians of Eastern Ontario and Climate Change. And then we're going to talk a little bit about some of the work that Matt spoke of yesterday, the FINS work and the wonderful partnership between Akwesasne and the River Institute, and some eDNA work that we've been working on. That was a prairie warbler, by the way, that I took. It was in a hydrocut, so a male prairie warbler. I have a big lens. So here he is, or she. This is an eastern fox snake. Unfortunately, we don't have them here, but they do occur in Ontario, and they occur in southwestern Ontario, so around Windsor up to the city of Simcoe, if you know southwestern Ontario at all. And they also occur up on the eastern shores of Georgian Bay. So they are an Ontario snake. And originally, if you just look at eastern fox snakes, Canada actually houses 70% of the global population of this species, and so has disproportionate responsibility for ensuring its conservation into the future. One other thing, we know that we have gray rat snakes here, and often people will say they're the biggest snake in Canada, and they probably are. But fox snakes are rivaling them in size, so a big fox snake will be 5 feet, 5.5 feet, and they're much kinder to you. I handle rat snakes a lot, and they like to bite. These guys seldom bite. The bites don't hurt. Just draw a little blood. They have tiny, tiny little teeth. I always tell people I've been bitten by squirrels, raccoons, parrots, many, many different snake species, and little children. And the kids hurt the most. This is the work of a wonderful PhD student who's now a consultant at Minnow Consulting, Jeff Rowe. So he did this work over a couple of years for his doctorate. And what I'm going to, that's a picture of two fox snakes that I took down at a sewage lagoon, actually in southern Ontario. And fox snakes occur in wetland habitats, which is one of the reasons I sort of presented them here, because we're talking about connections in aquatic habitats. And so historically, they once had a very broad distribution in southern Ontario and sort of north central United States. And because of a variety of threats that are well known for snakes and birds and everything else, habitat fragmentation, road mortality, and so on, there's been a diminution. But they used to exist in the plentiful marsh habitat, shoreline habitat, and tallgrass prairie. Southwestern Ontario used to have significant swaths of beautiful tallgrass prairie that's virtually disappeared. Fox snakes, unfortunately, also suffer from aficionados of snakes in tanks. So they're persecuted because they rattle their tails, even though they're not venomous, so people mistake them for venomous species. But they're also quite beautiful, so people used to follow us to try to use our knowledge to find snakes that they could sell in the pet trade. So you have to be really sort of clandestine when you're working on these species sometimes. And there's an emerging fungal disease that might influence this. So if you look at the map in the lower right, those are the regional populations we have. Imagine historically, pre-colonialism, European colonization, that would have been a much broader swath of southern Ontario, but now they're restricted, and this is very generous, to little pockets of abundance, down towards extreme southwestern Ontario, near Windsor, and up along the eastern shores of Georgian Bay. Before I dive into the study proper, I wanted to really put a face on the profound amount of wetland habitats that have been lost in Ontario. So this is a study that I hadn't been aware of until I was preparing for this talk. And what they have is an analysis of 200 years of change in wetland systems. So the dark spots here, it's not a road network. It's actually wetlands that existed in 1800 that have since disappeared. And so what this is a representation of how much habitat has been lost. And you can see in southwestern Ontario, it's virtually the entire area. All of the wetlands in those areas have been drained or filled or what have you. And this includes changing entire water courses like the Autonomy River. autonomy. But you can see Eastern Ontario also has a similar phenomenon in many townships. I'd like to make it a little bit more visually impactful by doing this. So this is a map of the townships. It's from 2010. It was prepared for Ducks Unlimited, and it's color-coded. So for each township, it's been coded with respect to what proportion of wetlands have been drained, primarily for agriculture, but also for urban expansion and road construction. And the red color means 85% to 100% of the wetlands have been drained. So there are townships where virtually every wetland that ever or once existed have been drained. And this is the milieu, this is the habitat where species of plants and animals are trying to cling to existence, and we're trying to prevent them from disappearing. And this is where fox snakes live. So Jeff and I were interested in fox snakes, and I have to admit that we started working on fox snakes because I love fox snakes, so that was really the spark. But then I began to learn about the ecology and the conservation challenges. And the first thing we wanted to do was to refine our understanding of their habitat preference. So for 2 years we did radio telemetry studies, 2 years of tracking in 2 different regions, and we looked at the habitats that they used and the habitats that they seemed to actively avoid. And then we took that information and created a mathematical model. If you're interested, it's called ENFA, Ecological Niche Factor Analysis. It's passé now, it's fallen out of favor. Don't use it, you'll have trouble publishing. But what we did was we took that information that we gained from radio telemetry and we projected it onto the landscape. And so we've made it fairly simple to understand visually. So we have unsuitable habitat. These are essentially impenetrable barriers. These are onion fields and cornfields, and fox snakes don't go into that. Marginal habitat, but the colors you really want to focus on are the yellow and red. Those are either suitable or optimal. And what you can see is there's virtually no habitat left in southwestern Ontario that is suitable, and especially optimal for the persistence of fox snakes in perpetuity. And the habitat that doesn't exist has been pushed to the periphery, and is found in conservation areas, national parks, hunt clubs, areas like Point Pelee National Park, Rondo Provincial Park, Lake St. Clair Wildlife Refuge, and some of the conservation areas that are held by conservation authorities. And so it really has been marginalized in this landscape. And so what Jeff and I wanted to do with our wonderful collaborators and a whole passel of students is understand what the connectivity issues were, whether they were still managing to maybe move along shorelines or to connect among these different seeming patches of suitable habitat. So we decided to do a genetic study. And so over 3 years, we went out and tried to catch as many fox snakes as we could, and we did very well. So we got about 600. Unfortunately, some of these are roadkills, but 600 genetic samples. This is non-invasive sampling, so we just take a little bit of blood from the caudal vein, and then we let these big snakes go, like 50 microliters. Very, very little. The triangles there are all of the different snakes that we caught and got blood from. So we have this. These are geo-referenced. We know exactly the habitats that they come from. And we wanted to understand genetically whether there was structure and evidence of isolation, demographic isolation, and whether we should be worried or not, or whether there was actually connectivity among these different patches of suitable habitat. So we genotyped them for using this high resolution method at the time called, these markers are called DNA microsatellites. They're very good at resolving differences between individuals, even closely related relatives, and fine scale geographic structure. They're also, or these data, by the way, are backed up with some pretty hefty genomics data that we have in hand right now. So whatever I tell you here is also been brought into the genomics age. So this is the picture. So we have very strong evidence for 8 distinct clusters. I've color-coded them on the map. So each of those color-coded individuals belongs to one genetic cluster and not to any of the others. There are a few ambiguous points, but it's quite striking. And what you can see here is that essentially each isolated patch forms its own demographic unit. It is isolated with not evidence of recent dispersal. So these little populations, unless we act promptly, are probably going to wink out of existence, because demographically, if you have a population of 30, 40, 50, 100 individuals, that's a pretty precarious persistence in a heavily impacted environment like this. I didn't want to leave you with just that dire message. So we've diagnosed what is going on here. We found some interesting things though. If you don't cut ditches down to the bottom and you leave them intact, fox snakes will use them. If you leave windbreaks intact, snakes will use them. If you don't turn your compost piles over, this is an egg-laying species, fox snakes will use that to lay their eggs. And so there are some relatively passive things we can do that might actually connect or reconnect these populations or push the numbers up. And we have made these recommendations to people. So there are ways to try to mitigate this without actively sort of managing it or getting ex situ conservation populations. And people are very enamored of this species and working very hard to try to rectify this. So that's the first case study, snakes. Now I'm going to turn to frogs. We know that climate change is having profound and potentially irrevocable impacts on species distributions. And we know that many species of both animals and plants are on the move, but not all of them. So it's not easily predictable based on their ecology. But many species of temperate organisms are moving northward, and we have evidence of that. Many species of montane plants and animals are moving upward, and we even have evidence in aquatic systems that they're moving deeper as aquatic environments move. But it's complicated to prognosticate what's going to happen, and this was really what brought us into this. Now, this is work that was done in collaboration with the River Institute by a wonderful postdoctoral fellow, Dr. Bojan Chen, who's now at the Ministry of Natural Resources and Forestry. Based, I would say, in part on his association with the River Institute and us and the work that he did. A species distribution model is mathematically complicated, but I hope conceptually simple to understand. What you do is you take data on where individuals of a species occur, both data that naturalists and amateur biologists report, but also from professional biologists, however you get a species occurrence. And then you record the environmental information associated with that occurrence and where it putatively does not occur, called a pseudo-absence. And you basically construct a mathematical model to better understand what best predicts where they occur and where they don't occur. And that's all that a species distribution model is, really. And so you can identify the environmental variables that are most important. They can be topographical variables. You can incorporate other species. So it's a wonderfully flexible tool, and this is the tool that Bojan was using for amphibians. But the interesting thing about species distribution models is once you've developed it, you can project it into time and space. So you have a species distribution model, you have a mathematical understanding of its relationship to the environment. And what that means is in areas that have not been surveyed ever, you can make educated guess. It's a little bit more than an educated guess, an informed a prediction of where you should be able to find these things. But it also means that if you have sufficient data, you can project into the future. So you can ask the question, what will these species distributions look like in 25 years, 50 years, 100 years? It gets a little bit dicey when you say, how about 1,000 years? But we can do this, and this is exactly what Bojan was interested in doing with amphibians. So I do a lot of amphibian work, and I'm very interested in Eastern Ontario amphibians and what's going to happen to them under climate change. You will know that there are various climate change models out there that are based on different emission scenarios. So what Bojan decided to do, or we decided to do collectively, was to take the most optimistic scenario, and that is emissions stopped increasing in the year 2020, haha. Very optimistic, and so basically we have a stabilization, and then the worst case scenario, business as usual, growth increases, emissions increase. So we have the pessimistic scenario, optimistic scenario, and we actually can sort of lay this onto a landscape. So we can ask, what are the climatological niches of these different species, and then we can ask what it'll look like into the future. It's imperfect, but it's very informative. So I'm not going to go into detail on the math. I'm not going to tell you anything. Suffice to say that Bojan did this. It took a lot of time to grind through the numbers and to validate and ground truth this. But I'm going to show you 3 examples from his work. And I want to emphasize, we're also doing this for gray rat snakes. We have a paper that looks like it's provisionally accepted in Global Ecology Change, a new journal. And so this is a general sort of way that we're approaching these kinds of issues. So this is, I'm going to say it, one of my favorite frog species. So we have my favorite snake, bird, now frog. This is a mink frog. Mink frogs do occur here regionally. Fred will know mink frogs well, I think, from where you are. And they're a cold water species. And they do extend all the way up to the James Bay lowlands. So if you go up to the sort of taiga, you can just almost all the way up there, you can find mink frogs going fairly far up. And so what the upper left is a species distribution model. What it is doing is color coding the habitats that we predict are most suitable based on those occurrence data and the model, and those that are least. So the red colors and the yellow colors are suitable habitat for mink frogs, and the green colors are unsuitable habitats, predicted based on the model. And these can be ground truth and verified by looking at where the individual mix will occur. And so you can see that in the present day area of Eastern Ontario, I should say this is Eastern Ontario and Northern New York, that the mink frog looks like that there's a huge range of suitable habitat. But what you have on the right is the same area, but it's what it's going to look like with the same color coding. So red and yellow is suitable and optimal habitat, and the green is unsuitable habitat. We have at the top row the optimistic emission scenario, and the bottom row is the pessimistic emission scenario. And then the first column is 25 years out by the year 2050, and the second column is 75 years out. And what you can see is that for mink frogs, it's not looking particularly good in Eastern Ontario. Remember, it is a cold water species, and so with climate change under any emission scenario, even in just a few decades into the future, it probably is going to disappear locally. I want to add, and I'll return to this, it may be that the range is simply shifting northward. So we're not talking about global extinction here, we're talking about regional things. I'd like to present a little bit more optimistic species now. So this is the northern leopard frog. I like it, not one of my favorites. It's a good frog, but it's a habitat generalist. And what you can see in the upper left, again, very broadly distributed in Eastern Ontario and Northern New York State. And what you can see that regardless of how many years into the future we're projecting, 25 years or 75 years, or whether it's the optimistic or pessimistic scenario, you're still going to be able to take your kids out to look for leopard frogs. And so that's a little bit more of an optimistic result. And then finally, this is a two-line salamander here. Again, we see sort of a broad swath of suitable habitat in the region. Two-line salamanders are interesting because they do not have lungs, and you probably knew that. So they're in the family Plethodontidae, and they breathe primarily through their skin, but also, interestingly, through their buccal cavity. And that means that they're associated with aquatic habitat, so if there's any prediction of a change in precipitation regime or increased temperature, then it could negatively impact a species like this. They're usually found near streams, riverbanks, or those kinds of persistently wet habitat. And again, we can go up to the predictions here. So 25 years out, under the optimistic scenario, 75 years out, the two-line salamander is going to persist, especially in northern regions of Eastern Ontario. But you can see there is one scenario, pessimistic scenario, 75 years out, where it looks like conditions for the two-line salamander will no longer be available in this region. So this is just a way, sort of heuristically, of understanding what might happen to the constituent biological diversity in this region. Bojen actually put this together, and if you attended the symposium last year, I think you might have seen this. This is 6 different species, and he's putting it together. So it's color-coded again. Warm colors are high species richness, so all 6 species is red, and the cold colors or white is no species at all predicted. So that's what we see now. That's a distribution of species richness based on these 6 species. And then what you can see is under these different scenarios, we are going to see a diminution in diversity of the amphibians that are native to this region now under climate change. So it's pretty robust predictions that we're going to see a dramatic change in the herpetofauna of the region. But I wanted to tie it together, and I wanted to perhaps embed this in some optimism with respect to action. So first of all, in an area like A2A or an area like the Frontenac axis, aquatic and terrestrial habitats must retain connectivity. The whole watchword of A2A and the Frontenac Arch Biosphere Reserve is connectivity and maintaining these forests. And for both aquatic and terrestrial environments, connectivity and particularly also contiguity to facilitate range shifting is incredibly important. It's also important because we want to retain the populations that we wish to persist in this area. But we have to start thinking four-dimensionally, if you like. We have to start thinking of time, too, in our planning for reserves. And these kind of models allow us to do that. The other thing I want to mean is, I wanted to say was, we're only looking at the species that occur here. We're not looking at the species to the south of us. So what you might see is a shifting of some of the species. So just like black-legged tick has arrived on our doorstep and brought Lyme disease, which I recently encountered, there are other species that are not as pernicious as black-legged ticks that are going to potentially move into our area. Things like tufted titmice, which are increasingly frequent in Kingston, and other species like that. So this is the last study I'm going to do. This is Stafford. He's about to defend his PhD in 3 weeks, month, I don't know. It's been finalized, and I agreed to the date. Stafford is from Tian, did a PhD with me, working with environmental DNA along the St. Lawrence River in his home community of Tian in the Bay of Quinte as well, but is also working with local people in Tian on Indigenous knowledge and changing perceptions of aquatic environments, and particularly fish. And he's woven this all together in a really nice PhD, I would say. But I'm going to talk about environmental DNA. We heard some lovely work that was being done yesterday in environmental DNA, so I'm just going to jump on that bandwagon a little bit and talk about some environmental DNA. So you'll know environmental DNA is increasingly being embraced by governmental organizations, NGOs, and academics to monitor biological diversity. A couple of decades ago, it just seemed like a fanciful notion, and now it seems to be adopted by lots of people as an affordable and logistically tractable way of tracking diversity. It relies on the notion that as all of us sit here, I am breathing in your DNA, you're breathing in my DNA. I'm breathing in more of your DNA collectively than you're breathing in mine, because I'm one, you're many. But when we're in an environment, any organism, you will live the signature of your presence by shed skin cells and other means. So DNA is present in the environment. In an aquatic environment, it lasts 1 or 2 weeks, maybe up to 3 weeks. And so what you have is a snapshot of the biological diversity that's sort of rolling through time. When you get into sediments, it might last centuries and even millennia. There's some eDNA work that's recreating environments with mastodons and things like that using permafrost eDNA. So it potentially can project back in time, but that's a different matter. All that we need to know is in an aquatic environment, we're getting a snapshot. So if we take a water sample and come up with some cool method to detect species, then we know what was there, more or less, at a given time, at a given place. And we can use that to address some interesting questions about conservation and biological diversity. We know in systems like the St. Lawrence River, there are increasing numbers of invasive species. And one of the challenges has always been in a massive river like the Yangtze River or the Nile River or the Mekong River or the St. Lawrence River, is they're very hard to monitor comprehensively with the money that is often afforded us by different governmental agencies. So the St. Lawrence River has over 80 fish species. And if you expand it down into the Gulf of St. Lawrence, of course, that number increases dramatically when you get into brackish species and saline species. And it's been heavily impacted by human activities, by colonization. So habitats vary with respect to the different levels of human impact. And certainly this is highly evident between Cornwall and Kingston. There are some areas where it's essentially agricultural land or industry right down more or less to the water's edge, and there are other areas that are more or less intact or at least have intact forests. Currently, fish monitoring relies mostly on gill netting, seine netting, and electrofishing. And I want to add that when we talk about eDNA, we're not talking about removing the need to do those kinds of studies, but rather we're talking about a complementary method. And I want to make that very clear. Stafford was interested in applying DNA metabarcoding, which was spoken about yesterday. And DNA metabarcoding is this powerful method where we can focus on a single gene fragment and using some fancy schmancy polymerase chain reaction, high throughput sequencing combined methods, we can amplify that target DNA for all of the different species in a particular group. So there's a marker called myfish, and we can use myfish to assay for the presence of all of the different species, and that's the method we're using. So all you need to know is metabarcoding gives us a ton of data, and it tells us whether a species of fish is present or absent with some caveats. So can eDNA provide a more robust assessment of fish diversity than traditional methods? Or how can it complement them? Can eDNA detect human impact on fish populations? So this is another sort of core question. And can we detect at-risk and invasive species with eDNA? And does it do so at a higher rate than we might expect by Siena? So this is the wonderful work of Matt and his team in the Fish Identification Nearshore Survey. This is a collaborative project co-led with the River Institute and, of course, the Mohawk Council of Akwesasne Environment Program. And this is a rolling, long-term study that is giving us some amazing insights into fish diversity and environmental variation in the Upper St. Lawrence River from the island of Montreal all the way down to Prince Edward County, the eastern terminus of Lake Ontario. So I wanted to highlight that. And so the sites that you see in that rather washed out map are all of the sampling sites that are represented in the data set that I'm going to talk about very briefly. And at each site we have seining data, so we know what fish are there based on the enumeration from seine netting. Matt's team also does drone work, and they assess water chemistry, substrate, like we get a whole host of different data. But they also took water samples. They took a liter of water samples using sterile methods, filtered it, and then we get the filters and we do our magic stuff in the lab with DNA metabarcoding. And we've been doing this now for about a decade. Not this stuff, but eDNA generally. So this just sort of summarizes it. If you're interested, Stafford just put out a paper co-authored with folks from the Environment Program, from the River Institute, and some other amazing collaborators. None of the projects we do are done by like me alone. I'm the pencil pusher. I look for money and I sign things. And I read things too. I do a lot of reading. So this is just a generic public domain photo of seining. That's one of my previous PhD students who did eDNA on turtles, out doing some water sampling. So this is the paper that resulted and contains some of the results I'm going to talk about. Unfortunately, it's a bit washed out, but I hope you can see there's a line at the top. So the first question we really wanted to tackle was whether eDNA was actually detecting the same number of species, the same species, more species than traditional seine netting. And this is called a species accumulation curve that some of you or many of you may have seen. So on the x-axis, we simply have the number of water samples, or if you like, number of sites that we have, going from 1 all the way up to the maximum number that are included in our study. And the number of species detected is on the y-axis, or the vertical axis. And so what you do is you resample many times. One site, well, in one site you don't resample, you just take the average across. But you know, if you have 2 sites, you resample 2 sites multiple times, and you see how many species you detect, and so on. And you get these very cool curves, and they approach a plateau, which means you're starting to get to the point where you're detecting all the species you're going to detect. Now, the top species accumulation curve in green, in faded, almost invisible green, that one, that's eDNA, and then on the bottom is seining. So as you increase, you see an increase in the number of species detected for both methods. But what you see is that you detect more using eDNA. And ultimately what we did is we detected 67 species overall after some fairly stringent filtering and bioinformatics, and with seining 38. So we detected almost twice as many species with eDNA as we did with seining. Most of the species detected with eDNA were also detected with staining, but I hasten to add there were some species detected with staining that eDNA did not detect, which is one of the reasons for using the complementary methods. You get slightly different pictures and you combine and you get a better picture. For eDNA, 9 species at risk and 5 invasive species. For staining, 3 species at risk and 2 invasive. So it detected greater number of invasive species and greater number of at-risk species. Importantly, if you detect an at-risk species, of course, this allows you to go back to the site and maybe investigate further. So often eDNA is the first clue that something is present, and you go back and you do some really good gumboot biology to understand it better. One of the other things that I didn't bring a graphic for that Stafford did was he compared sites where the coast, the shoreline was highly disturbed, where it was moderately disturbed, and where it more or less had intact functioning marshlands and forests. And only with eDNA data did he detect an impact. With staining data, we did not find statistically significant differences. With eDNA data, we did, that the intact shorelines had higher species richness. I wanted to do it a little bit differently here, so I chose 5 species, 2 well-known invasive species and some native species. And I wanted to bring it back to connectivity. That previous slide really wasn't talking about connectivity. So here we have from Prince Edward County, so eastern terminus of Lake Ontario, all the way down to Lake St. Louis. And this is combining the data from either staining or eDNA together. So if it was detected with one or either or both, it's a hit, it's green. And if it's not detected by either, it's white. And so what you can see with something like round goby, almost every site we detected round goby, which is unsurprising, a very common invasive species. And that does speak to connectivity and dispersal. So as the St. Lawrence gives, it also takes. It means invasive species can spread very rapidly, especially if their ecology is a fecund one and they have high dispersal rates. But what I wanted to look at is the stickleback, the brook stickleback at the bottom. And what you see, interestingly, is clusters of sites where you find detections. And what this means, in my mind, and I haven't dug into this as deeply as I'm going to, is that there are contiguous sites with suitable habitat that sustain local populations of this species. So there's ecological connectivity, but it also means that there's genetic and demographic connectivity, that there are adjacent sites where individuals can disperse out of, and you probably know that dispersal is an important part of the ecology of almost every species. And so this also speaks to the importance of natural connectivity among suitable habitats, and something we should pay attention to when we talk about restoration. So eDNA is very powerful for giving us these kinds of data where we can start to address. Now, 3 folks from our lab are here, and they're going to speak about various facets of environmental DNA, Alan and Bailey and Dilini. So I encourage you to attend their talks later and you'll get a more detailed picture of this. But I wanted to focus on this one. This is the tench. The tench was brought illegally into Quebec, and as in all fish, it seems that are in captivity and not really ever intended to be allowed to escape. It escaped, I think, in 1986 into the Richelieu River in Quebec, made its way to the St. Lawrence River, and since then has inexorably been making its way upward. And it's making its way up the St. Lawrence towards the Great Lakes, and it will be interesting to see how it alters ecosystems once it reaches the Laurentide Great Lakes. This is eDNA. This is from 2021, so these are data from Stafford's work. Dilini's going to talk about some of the work she's been doing. She's updated this, so we have it from 2024 now. But these are where eDNA detections came forward. So this is after filtering, after deciding how robust the evidence is. And we found a detection of the tench upwards, sort of around Brockville. And the point of this is that we found eDNA detections where it had not been captured. So eDNA perhaps can serve as an early warning system for invasion fronts of species that are just making their way into ecosystems. But what I want to emphasize is environmental DNA is a powerful tool where we can look at the effectiveness of restoration. We can track populations of not just invasive species, but species at risk, and can gain a greater understanding of the ecology, distribution, biogeography, and conservation of a whole host of species and entire ecosystems. So that really is what brings me to the end here. I haven't been looking at a clock, I should have been. So I'd like to try to tie it all together here. Human impacts on natural systems continue unabated, more or less, and they're going to continue. But we do have an opportunity to try to mitigate some of these, to try to counter them. And symposia like these are incredibly important in trying to do that. Humans and ecosystems are connected across spatial and temporal scales, and that human connectivity I spoke of initially is important to embed within considerations of natural connectivity because they are intertwined. Species ranges are changing, the distribution of entire ecosystems are changing, the nature of ecosystems is changing, and we have to embrace this, understand it, and decide what to do, and we need good information. And we need continued research, outreach, and conservation policy and action. So we need continued research by NGOs like the River Institute, by the Environment Program of Akwesasne, by government agencies, by amateur organizations. A lot of naturalist clubs undertake projects like bioblitzes and stuff. Oh, there is a clock, it's right there. I'm on time, apparently. Beautiful. If you're a professor, you better stay on time because the students will just leave anyway. So, but I can't emphasize enough the importance of really grounded research in communities, actionable research, and things like that. If given my druthers, I would just go stand in a swamp and listen to the birds and catch frogs and stuff like that without any sort of tangible outcomes. A lot of the research that I originally started doing was to understand where species come from, but I shifted because I wanted to take the stuff that I was learning from that and try to turn it into impactful research. And one of the ways I did that was by partnering with people smarter than me or different than me, and I think that's really important. But I also started sort of embracing questions that I thought would actually give rise to actions that would have real tangible impacts, and I think that's incredibly important too. And having amazing students and postdocs is a prerequisite. So with that, I'd like to thank my lab. That's a group of us. Only about half of my lab is in that photo. I was at the Canadian Herpetological Society annual meeting in Ottawa. So we have a fairly large lab. Without my undergraduate students, my graduate students and postdocs, I don't think much of what I've been talking about here would have got done. So thanks to them. Funding agencies, a lot of the work we do is funded by NSERC, the Canadian Foundation for Innovation, and various governmental agencies. Parks Canada has been a very generous sponsor of our work as well. I was at the Queen's University Biological Station for a long time, for 14 years as director and 2 years before that as associate director, and that really underpinned a lot of the stuff that we did. The FINS team and the River Institute have been amazing partners for quite a long time now, and really are important in advancing what we're trying to do with environmental DNA. And Queen's, despite it having a thick bureaucracy, has supported me in various ways. But I'd also like to thank you for rising a wee bit early and coming to listen to me at 8:15. Thanks very much.
Great, thank you so much, Dr. Lougheed. It's really interesting to hear about the diversity of species you study and the diversity of methods that you use as well. I found that, really amazing. We have time for a few questions. Does anyone have a question?
So I'm not super familiar with the geography of Lake Erie, but in your map of the snake populations, the olive green population looked like it was in the middle of the lake. Are they semi-aquatic, or is there a bunch of islands there that you guys were sampling?
The latter.
Okay.
The latter. There is Pelee Islands and Kelly Islands. So there are some islands on the American side and islands on the Canadian side. That is the southernmost point of Canada, that little cluster. And in fact, it's at the same latitude as Northern California. So we actually have latitudes that are the same as California. Did you know that? No, I was excited to learn that. And there's a robust winery industry there too. And actually there's a bit of conflict on Pelee Island between sort of vintners and people who want to preserve natural habitat.
Thanks for a fantastic talk, really, really informative. I'm not a scientist, so I don't really know much about eDNA, but you did a really quick description of it, of somehow breathing in our DNA and then exhaling and I don't know. Could you just explain a tiny bit more what eDNA is and how
Sure,
it works?
of course.
Thanks.
Yeah. So it's actually a much more complicated topic than I represented. So let's first envision an aquatic habitat. So fish have mucus, a mucus coating, and within that there's DNA from sloughed off cells and things like that. And so when you get contact between water and that mucus, DNA will be released or cellular debris will be released into the environment. So that's one source. A lot of organisms are broadcast spawners, so they'll put gametes out into the water. And so sperm and eggs are also contributing. When an animal dies, when we die, our DNA remains intact for a period of time. And as the animal is decaying in the water, DNA will be released. Some of it will be debris, cellular debris, intact bits of flesh, and things like that. That also contains DNA. Some DNA will eventually be released, and so you might actually have free-floating little bits of DNA. So the sources of DNA are almost too numerous to count. In aquatic environments also, there will be little organisms like bacteria. When we get bacterial DNA, it's actually within an intact organism. Purists would say it's only free-floating DNA in a medium, that is eDNA, and the rest is a different compartment. But broadly, when we talk about environmental DNA, it's from carcasses, from slime, from frogs and fish, from pooping. When animals defecate, cells will come along with the poop. Urinating, all of that stuff puts DNA into the environment. It does mean if you're planning some criminal activities, you may want to hold it until you get home. But DNA is broadly all of that. In air, it's a little bit more refined, but DNA can adsorb to little particles. And so there's a whole new line of research that is actually vacuuming the air, and the proof of concept has been shown, and you can detect diversity sort of spatially, and it's just little particles that to which DNA is absorbed.
First of all, I want to say thank you for including a species accumulation curve. I'm glad I didn't put it in my talk, so I'm glad it didn't steal your thunder today for that.
I would have shifted.
But I was wondering if you could expand on a little bit the differences in terms of, like comparing samples of eDNA and a sample of like a SameNet survey. Are those kind of one-to-one comparisons in terms of effort, right? Like, does the amount of work that goes into one eDNA sample equal the amount of effort that goes into collecting one, SameNet survey?
So first of all, I'll speak to the one-to-one comparison. Yes, in the sense that they're sampling exactly the same location. No, in the sense that eDNA moves in the water column. So eDNA does not get deposited in a particular area and stay there because of currents, thermal gradients. It will move around. So for example, in nearshore habitats, I think on one site, Matt, we detected muskie. There weren't muskie in the shallows of this. This was something from the center of the river, which you would never get in seining. So it is comparable, but eDNA is casting a wider net, if I may say that, into the middle of the river. So you're getting a bigger picture or a wider radius of the survey. So that disparity in part may simply be that. So that's one thing. In the field effort, sampling water, we don't use the filters you use. We sort of DIY it a little bit to keep the price down. But taking a water sample is very easy if you have the sterile bottles. And the effort is actually in the lab in bioinformatics. But we can do that very quickly in our lab now, relatively speaking. So once you have a workflow, we'll call it, then it can be done fairly effectively and fairly quickly. But it's a matter of economy of scale. If you're just doing sort of local stuff, like when we started collaborating with the River Institute with Matt's team, we had to get that all up and running from scratch, and that took a very long time. But now it's a little bit more automated. And there are indeed commercial shops that will do this for an exorbitant price for you. So the effort is not in the field. That could be super quick, but it's in the DNA extraction and everything that comes after. But if you've automated it, and a lot of people are doing that, or if you have a workflow that's really well defined, then you can reduce that effort a lot. As I say, though, I think it's important. I'm a gumboot biologist too. The two go together. Like you know, eDNA in the absence of actually understanding the river is just information that means very little. So I always tie those things together, I think. Thanks.
All right, so unfortunately that's all the time we have for questions, but I think Dr. Lougheed, you'll be here all day.
I'm hanging out.
He's hanging out. Okay, so you can come up to him and ask your questions throughout the day. And then we now have about a 2-minute break, so you can kind of stand up, stretch, maybe grab a coffee, and then we'll be back in 2 minutes for our invasive species session. Thank you.
All right, everyone, we'll get started. We're already a little bit behind schedule. So I'm going to give you one more minute to get back to your seats. So welcome to session number 6, which is about invasive species and their impacts. What do we know? So it's a short sessions. We're only 2 in the sessions, including myself. So I'm happy to host the session. I guess it's time for me to start my talk. So my name is Cristina Charette. I'm a research scientist at the River Institute. But I'm going to be presenting on my postdoctoral research that I'm doing at the University of Quebec in Chicoutimi. So I'm studying, actually what I did is I integrated machine learning and stakeholder insights to understand and model aquatic invasive species invasion risks. So like you all know, aquatic invasive species is a major threat to biodiversity. Research has been done, and we know that they have an impact on biological communities, food web structure, ecosystem functions, and services. So it's really important to understand the patterns of invasions to better align our management strategies. So a way to do that is to do invasion risk assessment. So this is when you want to evaluate the likelihood that a non-native species will be introduced, will establish, or even spread in a new environment. And usually we look at environmental drivers. And we use different approaches, including species distribution modeling, which you heard about a few minutes ago. And when we do that, what we use data from the presence of aquatic invasive species or invasive species in general. We overlap that with environmental drivers, and we create a model. And with the model, we can build predictive maps. It seems super easy, but actually it can get quite challenging because first of all, invasive species and their introduction is super highly context dependent. It can vary on the species in question, or the location, or even the environmental drivers that you're looking at or considering in your research. And most importantly, those environmental data is actually often incomplete or not necessarily available or open source. So a way maybe to come around that is to think about pathways and vectors of introduction of invasive species. So this is basically, when an invasive species is introduced by means and routes, and this is referred as pathways and vectors of introduction. So pathways you can think of as bigger categories like commercial shipping, recreational boating and watercraft, aquarium trades, or even commercial fisheries and shipping and things like that. I think I already said it. But that kind of data is actually available most of the time. So an idea is to integrate that type of data into those risk assessment modeling. So with that in mind, that's what I did. So my objectives were to first identify and rank the key important vectors and pathways in aquatic invasive species introduction, and secondly, to map the invasion risks. So my study area is southern Quebec. So we focused the study on the province of Quebec, but southern part because this is where you have it's most populated. The province of Quebec is not only economically important, but also ecologically important. It has major waterways, including the St. Lawrence River, which connects the Great Lakes to the Atlantic Ocean. And it's a route for invasive species introduction. So it is a hotspot for introductions. So I think since 2002, there's been something, I think 87 invasive species or non-native species recorded. But there's no universal framework in the province. So that's why it's important to assess invasion risks at this location. So what I did, I created a framework to do the assessment. And this is a summary and overview of the framework. And I'm going to go step by step and explain to you a little bit more in details. So obviously the first step was to collect the data, which was challenging enough and time consuming, obviously, even though most of the data was available on open resources from the Quebec government online. So the first type of data I collected was some proxies of vectors and pathways of introduction, and that included about 66 proxies that I have included into my modeling. So this is related to commercial shipping and ballast water, human population and urban development, recreational boating and watercraft, commercial fisheries, natural dispersal, and also climate change. On top of that, I've also collected data on the presence of aquatic invasive species, which was also on open resources. And this included all the invasive species together, but also divided by group or taxon. So it included fish, mollusk, plants, crustacean, reptile, hydrozoa, bryozoa, and entoprocta. And I had about 54 species included in my modeling. So the second step, now that I had the data, what I did is I extracted that data. So I built a spatial grid over my study area. So my grid contained cells of 9 by 9 kilometers, and I calculated the surface area of those cells. And then I overlapped them onto my spatial data, and I was able to extract each proxies for each of those cells. So overall, I had about over 5,000 cells that I included, and that gave me a data set that I was able to play with after that for the modeling. But, like I mentioned, I had 66 proxies that I wanted to include in my model, but I couldn't just put them all because that would be overfitting my model. So I had to think about a way to select the most important or most influential proxies. So I decided to combine two approaches. So one focused on the statistical approach and one focused on the stakeholder knowledge approach. So for the statistical approach, I decided to use the machine learning feature, which is called Baruda. I won't go into details about that. You can come chat with me after. But basically, with some permutation and iterations, I was able to detect the most influential, the most significant proxies to include in the model for all invasive species, but also for each group included. And then for the stakeholder survey, the goal with that was to be able to include local knowledge, but also incorporated different areas of concerns. And I created a questionnaire that I sent to different people across the sectors, and they had to rank in order of importance the proxies. And with that, I was able to calculate an average weighted score. And then I did a random forest predictive models, which just quickly I was able to build a model with the selected proxies from the statistical approach. I was able to build also a model in parallel with the selected proxies from the stakeholder survey. And then I trained my model and tested my model, and I was able to apply the predictive models to the whole data set to create the maps. So I'm just gonna go over some results on the selected proxies. So this is just a top 3 ranked proxies that were selected via the statistical testing. And you see that the proxies actually differed by taxa. So across the taxa, you see that hydrographic cover was the most frequent top one, which kind of makes sense because these are aquatic invasive species. But secondly, you can see recreational areas was also important. And we see something a little bit different when we look at the stakeholder survey. We see this is the top 5 vectors. So we see the number of boat launch, boat cleaning station and cleaning method, ballast water treatment method, marinas, and ballast water discharge events. So these were included into the modeling afterwards. Now let's move on to the map. So what does it look like? So here you can see a map looking at all aquatic invasive species combined using the Baruda feature, so the statistical approach. And you have the invasion risk coded in color. So the light color is lower risk and darker color, the higher risk. And you can see, obviously, like the St. Lawrence River and the tributaries, it's where you can see the highest risks, and also around major cities in the province. And if you compare that to the stakeholder survey, well, actually, you see more localized hotspots of higher invasion risks, and that kind of highlights the secondary and maybe pathway of introduction. Now what I did, I also did the maps for each group. So this is an example for the fish. Again, we see the St. Lawrence River being a hotspot for invasion with the tributaries. And when we see the stakeholder survey, again, we see the primary route, but you see also localized hotspot across the province as well. And in the end, what I did is I calculated an average for each administrative regions in Quebec to just determine which regions, were the most at risk. And regardless of the approach, Montreal, Laval, and Montérégie were the ones with the highest risk. So that indicates that maybe the strategies and priorities should be going towards those regions. So overall, the take-home messages is that we were able to develop a data-resilient framework to assess invasive species risk assessment. And we combined 2 approaches, which turned out to be really important because it gave us different perspectives, highlighting a more primary pathway of invasion using the statistical approach, but also highlighting maybe the secondary introduction pathway with the stakeholder knowledge approach. And in the end, this framework is highly transferable to other regions, but also you can modify the resolution if you're interested in a specific area, for example. So with that, I'll take any questions. I think I have time. One.
Any questions for Cristina?
Thanks. So it's just a general question on the definition of invasive. At what point is something considered invasive? Because obviously, as Steve said, there were 2 kilometers of ice here before, so everything here is invasive on that timeline. So I assume there's a timeline component to something being considered invasive.
There's a different definitions, and I think it's still debatable which one is the best. I don't think there's like a really one for all fits all, but we say that it's a non-native species that were introduced and now has negative impacts. That's one of the definition, but some others have different views on that. Like you said, some species were introduced way back then, but in my mind, if they have a negative impacts, that's when I would consider them invasive. Thank you. All right. Thanks. We'll move on to the second and last talk of this session on invasive species. So I would like to welcome Dilini Abeyrama to the stage. She's from Queen's University, and she'll be discussing how we can track the spread of aquatic invasions without seeing them.
Hi, good morning all. So I'm from a postdoc from Dr. Lougheed's lab, Queen's University, and I'm going to talk a little bit about how we can track invasive species in aquatic ecosystems, actually without seeing them. And so before diving into invasive species, I just want to give a little bit of introduction to biodiversity. So what is biodiversity? Biodiversity includes all the extraordinary life on Earth, including the genetic diversity, which is the variety between individuals within a species, and then species diversity, which is the variety of numbers and abundances of between different species, and last, the ecosystem diversity, which is the variety between different communities, ecosystem functions, and everything in between a given given ecosystem types, like a desert or a wetland or any kind of ecosystem. And why is biodiversity important for us? So as you all know, biodiversity is providing us all the necessities for our life, like food, air, any other resources, and also it provides many ecosystem functions, purifying our waste and everything. So have we been treating it well? To be honest, no. And as Steve also mentioned in his talk, the biodiversity is at a critical condition, and over the last 50 years, based on the Living Planet Report 2024, the overall decline of biodiversity is around 73%. And in this graph, it shows like at each different types of ecosystems, how much is the average decline in biodiversity. So, and you can see that those numbers are really high, and it's really alarming. And so what is driving this loss of biodiversity? As Steve also mentioned, there are 5 main drivers for loss of biodiversity, and they are habitat loss, overexploitation, invasive species, pollution, and climate change. And based on the Living Planet report from WWF in 2018, you can see what each of these threats have, like how much effect on different taxa like birds, mammals, fish, and other herpetofauna. And then let's talk a little bit about invasive species. So what is an invasive species? An invasive species is a non-native introduced species that has the capability of spreading and adapting easily to this new environment, causing tremendous threats to native species in this local environment, and also bringing harmful effects for everything in the ecosystem, like human and all the other ecosystem functions. So in this heat map, it shows the global distribution of alien species, and so we need to remember that alien species are not the invasive species. Alien species are non-native species, but they don't have to be necessarily be invasive, but all the invasive species are alien species, and you can see that the heat map is like showing some alarming conditions. And globally, they have recognized more than 35,000 invasive species. And when it comes to Canada, there are more than 1,500 invasive species recognized in within the country. And to be honest, this number might be like even higher, that we are not that we haven't encountered all these species. And so among all the provinces and territories in Canada, Ontario has the highest impact from the invasive species, with about or like more than 441 invasive plant species and more than 191 aquatic invasive species. So these species are mostly found within the St. Lawrence Great Lakes ecosystem, mostly due to international shipping channels and multiple waterfronts, and also huge amount of import goods in the area. And in this graph from the Invasive Species Center, you can see that the cost of dealing with invasive species can increase rapidly when it comes from prevention to long-term control, because these invasive species are like diseases. If you are not controlling them or eradicating them at the beginning, it will be very challenging to control them, because they can spread like crazy. And the cost due to these invasive species is really high. Just in Ontario, the economic loss from invasive species is around 3.6 billion per year. So that's a huge economic loss. So therefore, to avoid all these negative impacts, it's critical to manage invasive species. In order to manage these invasive species, we need to monitor them. So to monitor invasive species, we can use traditional monitoring, which is basically looking for auditory signals as well as visual signals. But there are some issues with these, or like negative factors related to these traditional monitoring, such as labor intensive, less sensitive, and also we need high taxonomic knowledge to identify all these species correctly. Therefore, it is better to have more advanced tools that are more sensitive, that can provide us with some real-time data, and also especially non-invasive, so we are not disturbing the organisms or the habitat itself. So one such tool is environmental DNA, like Steve and also the last talk from yesterday mentioned. So environmental, in this figure, it shows how sensitive environmental DNA can be when compared to some traditional visual detections. The number of species of fish detected with environmental DNA is relatively high compared with the visual detections. So what is environmental DNA? Environmental DNA is genetic materials that is sampled from environmental resources or environmental sources such as water, soil, air, sediment, spider webs, and etc. And from where does these environmental samples get genetic material? So it can be from organismal secretions like mucus, gametes, feces, or some other tissue samples like hair, tissues from dead animals, or other tissues they are shedding in the environment. In this figure, it shows the four most used environmental sources for environmental DNA: air, water, soil, and sediment. And something cool with sediment eDNA is that when we look using sediment eDNA, it allows us to take a snapshot of the temporal distribution of different species, because with sediment we can date the sediment sections and compare like what time segments are captured in there, and when we compare that to eDNA signals, we can see when these species were detected in the history. So for our study, we focused on water as our environmental source, and our amazing field team from Finns and River Institute collected water samples along the St. Lawrence River from multiple locations. And at the Queen's University, we extracted, amplified, and sequenced DNA, especially to look at the fish diversity along the St. Lawrence River. And so what we saw. So we looked at all the fish species we could detected in these environmental samples. But for my talk, I am focusing only on a few invasive species that we detected. So we here in this slide, I am focusing just on one river section in our study, Lake St. Louis. So here, and also I myself did the extraction for fin samples from 2024, but we also compared our data with Seining, as Steve mentioned, and also Stafford, who is one of my lab mates, did this study for samples from 2021. And in order to get a temporal sense of this distribution of these species, we compared the data from 2 years for the two methods. So the green here in this heat map shows detections from eDNA, and purple Seining, and yellow for both methods, and white if they are not detected at all. So you can see how different the detections are. So a very common species like round goby were detected equally with both methods, and other species like less common species like tench or common carp, they were detected mostly with environmental DNA only. And some other species like tube-nosed goby we detected with both methods. And in this graph, which is a little complicated and very busy, I have all the six river segments we studied in our research, and same pattern. You can see that common species like round goby was detected equally with both methods, while less common species, common carp, goldfish, and tench, and also round tube-nosed goby here, were detected mostly with environmental DNA. So what we found, so you can see that compared to traditional methods, environmental DNA is a powerful tool to especially look at less common or rare species, such as the early stages of invasions. And as you know, not all the species are active equally at the different times of the day or equally during the whole year. So in order to get a proper understanding about their distributions, we need to conduct continuous monitoring throughout the year, maybe different seasons, and also, if possible, different times of the year. And as Steve also mentioned, to get the perfect idea about the progress or the patterns of distribution of these species, we need to combine powerful new methods like eDNA as well as traditional monitoring, because in the heat maps also you saw that some species were detected only either with eDNA or either with traditional methods, and sometimes with both methods. So in the perfect scenario, it's better to combine the two methods. And finally, I would like to thank all the people and all the institutions that provided us funding and help. And I'm happy to take any questions. Thank you.
Thank you so much, Dilini. Unfortunately, we're running a little bit behind, so if you have any questions for her, please see her after the next session. So we'll just move on to the next session right away. Sorry, no breaks. We're just trying to catch up on the schedule.
Hello, everyone. Good morning. I am your session chair for the Social Ecological Perspectives for Understanding Environmental Issues session. Apologies that we can't take a short break here, but we'll have some time for break soon. And I'd like to introduce our first speaker, who is Siena Margorian. And she is an amazing student at the Freshwater Institute at McGill University. So please give her a hand of applause to welcome her to the stage.
Okay, good morning. So I just wanted to first acknowledge that we're gathered on the lands of the Akwesasne Mohawk, and as a student at McGill University, Montreal is situated on the unceded and traditional territory of Ganagahaga. So, as we all know, there are social systems and institutions that attempt to interact with and govern ecological systems or ecological phenomena. And a way that we can conceptualize the interdependent and reciprocal interactions between social systems and ecological systems is social ecological systems and FIT, or SES. So there's individual actors like yourselves that take part in different organizations or institutions, and these will affect ecosystems, and as changes happen to the ecosystems, these will then affect the social institutions. So this is the kind of framework that we'll be thinking about this work. And I will be focusing on the American eel. So to understand the unique social ecological system of the American eel, it's really important to understand their life cycle. So all American eels start in the Sargasso Sea, which is about the area of the Bermuda Triangle, and they'll start off as eggs. They're born there, and then they'll become these kind of larvae or leptocephalus that are very transparent, which is really hard to see on this projector, and they'll travel hundreds or thousands of kilometers to upper coasts, and then they'll start to become glass eels, which are still kind of transparent, and then they'll start moving inland and they'll become a little bit more pigmented and become elvers. Then they'll become yellow eels, and then they'll live in freshwater for an undeterminate amount of time, and then at some point, for some reason that we don't entirely know, they'll decide to become silver eels and then take the long journey back to the Sargasso Sea, where they'll reproduce, lay their eggs, and then eventually die, and they'll start again. And what's important to note about American eels is they can't reproduce in captivity. Their life cycle can only be complete in the wild. And they have a very expansive range, all the way from Greenland to the Caribbean. So an extremely difficult social ecological system to manage and conceptualize because it crosses so many borders, different groups interact with them. So very, very difficult to kind of work together and manage the American eel. And what's really important in our Canadian waters is that the American eel in the upper St. Lawrence River and Lake Ontario are all females, and they're the largest and most fecund females of the entire species range. And it's estimated that a quarter of the egg-laying population is in the upper St. Lawrence and Lake Ontario system. So really important area that falls within Canadian waters. So my main question is, what is the social ecological fit of American eel conservation management in Canada? So I am trying to answer this through a policy analysis. I have begun with a database search. So I look through databases with like ProQuest or Google Scholar, and I'm looking up papers, and I'm picking specifically ones that are published in either English or French from the last 15 years, and that pertain to American eel management specifically in Canada. And then I will extract some information like what kind of management measures that they recommend, if there's like policy recommendations, any specific issues or threats that they highlight, and then, you know, the boring stuff like quotes that I'll pull out, or the citation, or the year that it was published, and things like that. So I'll end up with this really, really big database. And with all this information, I am trying to assess 5 dimensions of social ecological fit. And social ecological fit describes how the social institutions match with the ecological processes that they're trying to interact with, and how well they're working together. So these are some of my preliminary results. I still have some documents to go through, but social fit we'll start with is how the institutions interact with are managing or are meeting the needs of the rights holders and stakeholders that are part of their system. So I've determined that it's poor. And the reason why I have determined that is that populations are in pretty steep decline, and American eel is quite important to a lot of First Nations in Canadian jurisdictions. And declining populations is making it a lot more difficult for different Indigenous communities to engage in their practices. So there's been a lot of concern about declining populations from First Nations, but also from non-First Nations. As I mentioned before, eels can only can reproduce in the wild. They can't be reproduced in captivity. And if any of you had sushi, you might have had eel in your sushi. You would know that eel is very popular in certain cuisines. And this has made the price of alvers, which is predominantly in our maritime waters, to skyrocket. So in the past few years, $4,000 to $5,000 a kilogram. And so there is a very big and contentious, which I'll get to soon, alver fishery out in the Maritimes. And there's been a lot of tensions between First Nations and non-First Nations, between license holders and the DFO, but we'll kind of get into that some more. But yeah, a lot of it has led to poaching, which is also not the same as First Nations who are managing their fisheries outside of DFO management plans, but again, I'll talk a little bit about that a little bit more, but very contentious. So as you can see, the rights holders and stakeholders, not very happy. Next fit is ecological fit, which I would also say is poor for the very simple reason that populations have declined drastically. Some estimates have said that eel populations in Lake Ontario have declined by about 97 to 99%, and then overall in Canadian waters, about 50 to 70%. There was a report this year that indicated this. But this graph here, the Moser-Saunders Dam, which is right nearby, has an eel ladder, and they were able to count eels passing through that eel ladder for several years. So you can see starting in 1975, and you can see just how drastically the eels coming over that eel ladder have declined. So I would say that ecological fit is poor. Scale fit is how the ecological system is managing, or the social system is managing the ecological system as a whole in terms of the entire scale. This is a little bit difficult considering that the scale is from Greenland to the Caribbean, but speaking of Canada, we're kind of thinking of the system as a whole. So there's really inconsistent data throughout Canadian waters on American eel and throughout the life cycle. And the DFO intends to manage the fisheries through a federal provincial management plan. However, certain First Nations out east are exercising their Aboriginal rights to manage their own fisheries and are issuing their own fisheries licenses, so there's not always agreement about how it should be managed. And then it's also important to note that both federal and provincial listing decisions will affect management plans, so it's not quite the same. And in terms of vertical fit, which is how, when there's multi-level governance structures, which we have, how those are working together, how the different actors in that multi-level structure are working together, and that would be poor. COSWIC, which is the federal listing decision, which does not necessarily affect management plans, has listed the American eel as threatened. Although we're waiting on the Species at Risk Act, it's been about 13 years, and actually it's been so long that the old round of consultation has expired, that they've started a new round of consultation this year. Ontario has listed the American eel as endangered, Quebec has it as likely to be designated as threatened or vulnerable, and Newfoundland and Labrador has listed it as vulnerable, but there is no listing in New Brunswick, PEI, or Nova Scotia. And in terms of horizontal fit, I would say that it's good. Horizontal fit is how different actors within the same kind of multi-level governance structure are working together to try to collaborate, and there are a lot of groups working on sharing knowledge. So the Eel Knowledge Working Group, the American Eel Conservation Network, the Eel Passage Research Center. There's a lot of groups that are trying to share knowledge within one another and improve practices, which is lovely to see. So next steps for me in my research is to finish reading all those documents to continue to provide evidence for this policy analysis. And then I will also be conducting a case study with professionals working in the field of American eel conservation, and I'll be putting out a questionnaire and doing some interviews. So if you would like to participate, I have a QR code there for you to pass along your contact information. But I would like to thank my funders and my lab, and I'll take any questions.
Thank you so much, Sienna. That was so interesting. Do we have time for questions, Red? Yeah, okay, perfect. Do we have any questions?
Yeah, I think with a lot of these species that are in multiple countries, that there ought to be organized celebrations for when the elvers arrive in each province and state and country. And similarly with the upland sandpipers, that there should be a celebration when they leave for South America. And we have a neighbor, going back to the upland sandpipers, we had a neighbor who, he was a Dutch immigrant, but he'd been all around, and he called the upland sandpipers the Argentina birds. And so I think we've got to have public celebrations of these things in the way that the monarchs are celebrated when they go south and come back.
Absolutely, I very much agree.
I also agree, and maybe this could be, the spawn of an eel celebration here in Cornwall. Do we have any questions from the audience? Start with Dave.
I just wanted to see if you're going to include Bill 5 and Bill C5 into like your analysis, considering its implications on Sarah.
Yes, I will.
Great. Okay, we have one more question over here.
Am I right in assuming at Lakes St. Louis there is a good population of eels, and we're just not seeing it up in Lake St. Francis? Am I right?
Sorry, which lake?
Lake St. Louis? St. Louis?
Are you, so sorry, the question is there a good population, a healthy population in Lac St. Louis?
Yeah, and not in this area.
I think populations are just kind of struggling all throughout the St. Lawrence River, down Lake Ontario. Lake Ontario is particularly hard hit. I think Lake St. Louis have slightly better populations than Lake Ontario, but I think it's pretty poor throughout. Correct me if I'm wrong, Marian.
I'm just wondering if they're not getting from that point to this point for some reason. Is
Why
the
are they?
locks? Yeah.
Yeah, the dams are a huge barrier to them accessing their historical range and like their historical areas where they would live, but also the turbines are a huge cause of mortality for the eels. So that's another kind of point of contention between many groups is the turbine mortality. Yeah.
Thank you so much. Do we have any more questions?
Well, when we had a meeting with you guys, you were talking about the turbines, and we're talking about like what are like the options and like who needs to be pressured and stuff like that. I was wondering if you could talk about that in terms of, because you also talked about the percentage of eels, like that seems like the biggest issue when it comes to their endangerment.
Yes, so the turbines are a very big issue, again, very, very big cause of mortality, not only because passing through the turbine, so when they're going back out, they're migrating back out to the Sargasso Sea, they have to go through the turbines. There's a very high mortality rate, but it's also cumulative mortality. There's so many dams that they have to pass through when they're migrating back out. Some of the options, I guess the most obvious one, but potentially the most difficult one, is you remove all the dams, but yeah, exactly. The next option, there are certain companies who are coming out with like fish-safe turbines, but again, that's a really big endeavor to renovate the dams and replace the turbines with fish-safe dams, especially if you think about how, or fish-safe turbines, if you think about how big the turbines are, especially at Moses Saunders. I think that would be the next best option. And then there's also trap and transport programs where they'll catch a certain number of eels every year and then just move them past the dam and release them past the dam.
Great, thank you so much, Sienna. Could we have another round of applause for her. Thank So the next speaker, I would love to introduce Abraham Francis, who's going to talk about reciprocal healing amongst all of creation. And yet we have the pleasure of listening to him talk. So please welcome Abraham.
Thank you very much. I think I have slides, right? So I'll just kind of start, right? Sego sewa guego de holohiate yungyat swagani atrono the negani haga nena akuzos neganagale. So hi, my name is Abraham Francis. I'm Deer Clan and Mohawk from the community of Akwesasne, born and raised. And Yunkiat the Genhas is a word from the Ohandagalluwadekwa that I've really fallen in love with, that they support us. And I think that this for me is this offering to a challenge within the world of shifting paradigms, and particularly one emerging from a Haudenosaunee paradigm. And what does that mean to the landscapes here? What does that mean to the work that we're doing? And trying to figure out the best way to articulate this. And a bit of this comes back to one of these foundational pieces, and this is actually a chapter in my dissertation that I'm threatening to finish this year. And I think it's changed a little bit, but it still has these core tenets of a rejection of the concept of environmental management, at least sort of the foundational principles in which they are founded. And that's what I've spent a lot of time learning over these years. And so this for me also is founded and grounded in this concept of Jinigoli Hodo. And it's that we are trying to figure out a way to care for land that is consistent with our biocultural framework, how we relate to the world around us and how that is conceptualized. And for me, this emerges from who I am and where I come from in a deep understanding of how that is connected to a larger Haudenosaunee relationality with the world around us, and how there is a bit of nuance across the nationhood. Now, hopefully and so this again is addressing this larger question for me of how can we care for creation as a reflection of the people of those lands. And so for us in here and within Haudenosaunee territory, this emerges from the Ohandagalluwadekwa. It would look different for the Anishinaabeg, the Mushkowagowik, all sorts of different people with different relational governance. We all emerge from these landscapes in very different ways. And also, what I'm really attempting to do is reject the concept of the imagined Indian. And this is a term from Visner's work around how we are perceived as indigenous people. You know, the idea that we're out touching trees and talking to them. And I always love to add, like, if I am, it's none of your business. But, you know, and I think this is such a perfect picture of this, like Iron Eyes Cody, this famous part of movement back in the day where he would see pollution, and I actually think it was pollution in the Great Lakes, and he would start crying. And what I love about this story and how hilarious it is that he turned out to be an Italian guy. And it just makes me laugh to think about how this implicated us as a people and the perception of the world of what we have to offer within this larger dialogue around environmental caretaking, around climate change, and these global issues that we're trying to push back against. And so it does address some of this dimension of the way that pretendians can impact the work that we do and ultimately affect the way that we occupy space within institutions and the work that we're producing. And I continue to meditate on this because, you know, some of my favorite work was actually created by pretendians, and I gotta renegotiate what that means to me. And so, you know, as I continue to sort of work my way through understanding the relationship of indigenous people within climate change, a lot of this to me kind of comes from the absence or the mischaracterization of who we are. The way that we see this global effort of indigenous people, local communities that disrupts the way that indigenous people's law or representation within international laws are weakened through a dynamic of that type. Or the way that we are misrepresented within the social development goals. We only sort of show up, it seems, within, you know, peace, justice. And it's like we are a holistic relational people, and we should be spread across these SDGs. And I'm seeing First Nation communities attempt to reimagine these within that in a way that is consistent within their biocultural framework. And so, you know, for me, spending my journeys with environmental management, I always feel like there's such a new sexy term coming out for environmental management, and the new way that some individual and some government has come up with a solution. And, you know, in particular, one of the more consistent ones for me within an indigenous frame of thinking is adaptive management. This sort of continual process of learning and adapting, and specifically that being one of these good strategies for environmental caretaking, but still sort of misses the mark in these certain ways. And a little bit of this for me comes back to the historical context and within which this exists, the value systems in which a lot of this work actually emerged, the way that environmentalism as a movement emerged from the past, you know, and a bit of how we became distanced from the land, and understanding that dynamic in the way that indigenous knowledge and indigenous people's knowledge was undermined and not canvassed. Now we do see it be coming into the conversation more, but it sort of comes in this sort of like, I almost want to describe it as fluff. I want to see more action. I want to see the materiality behind this. I want to be engaged in those ways. And that's what really draws me to this work, and a rejection of some of these concepts, because a lot of these concepts emerge from a place that imagined indigenous peoples as are basically irrelevant to these landscapes, that we weren't actively caring for these landscapes around us, as if our relationships to these landscapes were not significant or we didn't have something to offer. Now you might hear the like 85% of biodiversity is located on indigenous people's land, which is a really exciting term and is actually wrong. There's a much more complex framing and relationship, and it's really important that we don't sort of, we question things and where they emerge from and how they impact the work that we're doing. And so this is my attempt through Yunkiat Tegenhas to challenge that. And this emerges from, as I've said, the Ohandagaliwa Dakwa, our speech that we give before and after an important decisions are made in meetings, in an attempt to acknowledge all of creation and keep them in mind as we are moving through our space. And so this is the foundation. This is who I am, and I think about this consistently within this work, and then think about my roles and responsibilities to creation and the world around me, and how I can layer this with different teachings within my culture, whether that be the Turo, or that be the Guyana Goa, the Great Law, and bring in Skana, Gasustasla, Gatniguhlio. And this is sort of related to like the rest of it in my dissertation, but a lot of this emerges from an understanding of biocultural context and the way that our culture and our language and the land are so intimately tied. And for me, this sort of emerged from an understanding of the way that environmental violence within this territory implicated my people. And then also sort of this longer and more in-depth exploration through forest stewardship, and how forestry and the forest represent so much to us as a people, and are actually sites of restoration, are sites of transformation, and inviting that within this idea, within this theory. Now, a lot of this too emerges from this rejection of fitting things into these neat little boxes, and this emerges from my love and celebration for Carol Warrior, and this infamous quote that I'm always carrying around, introducing into spaces, but in particular emerges from this meditation on fluidity and fixity, and the way that we try to control things and fit them into these neat boxes and how they reject that. And I think even the eel is a perfect example of the rejection of these little rigid boxes and how these borders actually make no sense to us. And in Akazasne, how we directly reject this through living our lives on a daily basis. And this is just one of my like favorite pieces, is this recognition of cycles, form and life and birth and death, and this relationality across those pieces. You know, and this ultimately sort of relates back to my understanding and my articulation and relationship with the rights of rivers, and this growing I've been on. And honestly, what I've spent the last yesterday and the day before gathered with over 120 First Nation community members talking about what does this mean as our opportunity to reject Bill 5, Bill C5, and the various threats to our rights and our relatives, and an honoring of Josephine Ba, who really brought to life water is life. And this speaks to that importance of that environmental restoration needs to be accompanied by cultural restoration. The environment is healthy and nobody knows it, then what are we going to do about that? And then also a celebration of the river strategy and the ways that we're trying to gather and we're trying to articulate movement and community together, and these various ways that we're establishing and situating that through our roles and responsibilities of allies, consistently coming back to these roles and responsibilities. And I think of this theory as something that can help us sort of connect, you know, these various various social eco spaces that we work across, from government, First Nations, you know, relatives, who knows. And, you know, this to me sort of is the celebration of a lifetime of learning. This theory emerged for me with a couple of frustrated grad students, and I'm like, I hate stewardship. I hate that word. It's so not working for me. And that emerges from like Henry and Peggy's mentorship. And then in grad school, through Teresa Beardall, she helped me say, just write your own theory. And I was like, oh, you can do that? And so that's what I did, and I've been enjoying that. And this sort of is that continual celebration. And this is, you know, going from cleaning up the St. Lawrence a long time ago and a lot skinnier to now to being able to continue this work. And, you know, it speaks to this, again, that restoration and the ability that if we're able to be back in engagement and a good relationship with the lands around us, then there will be better ecological outcomes. But not just that, community impacts. Like, I imagine a healthy environment accompanied by healthy communities, a rejection of capitalism, a rejection of this landscape we're living in that it promotes hyper-individualization, excessive work, into a more gentle, kinder landscape for us. And it, you know, I am still writing it and finding out like the edges of it. So I really appreciate you guys listening. My gratefulness to everybody here, my gratefulness to everybody that spoke into this. And yeah, so appreciate the opportunity to share Yung Kiat the Ganhas with you. Tuknigo.
Is the mic on? Yeah. Do we have time for questions? Do we have one question? No. Getting my work out here.
Thanks for this great talk, Abraham. Just quick question. I'm like really interested in words and our relationship with words, and I'm just genuinely curious, like if you could expand a little bit more on your issues with the term stewardship and why.
Yeah, so mainly because a steward is a concept that was emerged from Europe and is a relation of like the fiefdom and the relationship with the king and, you know, the land that they let basically peasants work on. And, you know, this idea emerges from a landscape of everybody's equal here. All of creation is equal. We matter just as much as the eel, and the eel's presence here matters just as much as ours, and we need to go from that place. You know, and I think it is like an attempt to reject a bit of where private property emerged from too, because a lot of this sort of issues and concerns around the commons, right? I'm trying to remember the exact one, but my brain is so tired. And that here in this landscape, we have the one dish, one spoon as a rejection of that concept. That it's through community and understanding and story that we were able to care for these landscapes. And so when Europeans arrived, they were like, oh my God, there's no people here, which, by the way, had to do because we weren't Christians. But, you know, the expanse of this implication we feel today, even 500 years later, right? So that's sort of the intentionality I'm trying to put in here, but also prioritizing my people's language over English.
We don't have any more time for questions, I'm so sorry. But thank you so much, and could we give another hand to Abe? And I'd like to introduce us again for the next presentation. So I don't know if you want to take this. You take that one. Okay, so we'll just wait to get our slides up, but Abe's going to kick us off here, and we're going to talk about eels.
All right. So me and Marianne had wonderful conversations about the eel and our frustrations around what is happening and this long history. And so we're trying to the tale of the eel, a new dawn, and thinking about all of the transformative work that we've been up to over these days. And do you want to add anything to that, Marianne?
Just I feel so grateful to get to work alongside Abraham, and yeah, I'm excited to present some of the stuff we've been working on for the past couple years anyway, and longer for thinking about it anyway.
Exactly. And so, oh, geez, I went a little bit far. And so we have been working on these community reports, and in particular, these community reports that were showing the eels that were piling up on the north side of Cornwall Island, Gawanoge. And that this sort of started this really big effort in this area by bringing visually the impacts of the turbine. And in particular, building on a long history of violence from the seaway and the dam development on our community, and this just being a continual violence. And we thought it was important for you to see these piles of eel. And I think something that I always come back to is the words of Stephanie that told me that she spoke about how there's something so intimate and exposing about their white bellies. And to see that within this imagery, and how actually the eel is designed for camouflage. And in this moment, this is like a cry for help. And I really appreciate that framing, and I think about this work from that place of that role and responsibility to step up and speak up and do something about this issue. And for me, this emerges from that space of eel as relative, right? I talked about Yonkiat the Genhas, and to me, this is an extension of my role and responsibilities to speak to a relative that is threatened, and to stand up, because losing that eel is a loss of language, it's a loss of values, it's a loss of medicines. It makes us more susceptible to political violence, to environmental violence. We are more susceptible as a people. And ultimately, it connects to this larger piece of if we don't acknowledge creation, we don't care for it, then it will leave us. And I sort of feel that there is this situation that is emerging in this moment through the eel's story. And this sort of connects to this larger effort that we've been working on for years through the River Rapport, is this holistic ecosystem health report for the Upper St. Lawrence that is framed within the Ohandagalua Dakwa. And this gift from this sort of idea gift from Henry Lickers that I have had the lovely opportunity of working with so many wonderful people at the River Institute on dreaming and challenging the way that we do science, and we write about science and we talk about it through the River Rapport. And this sort of comes back to this sort of core thing that we always keep talking about, which is now that we know, we have that responsibility to do something, and the power in that knowledge. And this is ultimately what connects to this larger conversation that I am now going to kick over to Marianne to add more dimension to.
Thank you so much, Abe. Thank you. So just to give some more context to this ecosystem health report, it's called the River Rapport. I'm the scientific lead, and Dr. Lee McGaughey leads the project. And we work closely with our friends at the Mohawk Council of Akwesasne, and I co-create everything. Everything we do in the project is co-created with Abraham Francis. So I am behind my computer, all day, looking at data, looking at reports. We have 35 ecological indicators that were nominated by the community. So the community wanted to know more about them and how they reflected the health of the Upper St. Lawrence River. So I'm not going to go into the details of this slide that has very tiny writing on it, but just to tell you that each of these indicators, myself and my team go through a standard operating procedure. And it's a procedure used to identify all relevant data in this case on particular subjects. So as I mentioned, we have 35 indicators, and eels is one of those indicators. So we look through academic literature, we look through government reports. What I'm trying to do is compile all the information on American eel in the Upper St. Lawrence River, crunch that information and make it accessible, and engage the community, mobilize that knowledge and translate it so the public understands what's going on in their backyard. So for the eel, I've been working for the past year and a half on it, and I've went through 630 papers and reports. And it's been exhausting, to say the least, but it's been really insightful. And as Abraham mentioned, the more you know, the more responsibility you have. So looking more at those 630 reports, breaking it down, this is who holds information about American eel in the St. Lawrence River. So you can see here, so this is just a bar graph, and the red is information that is not accessible online. So it exists, but I cannot access it. The yellow is information and reports that is somewhat accessible, so you need a subscription to be able to download it. So it's not accessible to the general public, but it is accessible to university students or basically any academics. And the blue is information or reports that is easily accessible, so I could just go online and download the information. And if you look at how this information is broken up between the different parties here, industry holds the most information on American eel in the St. Lawrence River. And this industry is hydropower dams. So on the Upper St. Lawrence River, we have two major hydropower dams. We have the Moses Saunders Dam and the Beauharnois Dam. And these are operated by New York Power Authority, Ontario Power Generation, and Hydro-Quebec. So they hold most of the information on eels, and most of it is accessible, and there is a portion of it that isn't accessible. Government holds the second most amount of information. Academics hold the third most. And if we break this down further by industry, so as I mentioned, there are three different industries that operate the dams in the Upper St. Lawrence River. So if you look at those three industries, so the Moses Saunders Dam is owned on the Canadian side by Ontario Power Generation, and on the American side by New York Power Authority, and the Beauharnois Dam is owned by Hydro-Quebec. So you can see here that Hydro-Quebec is doing great. Like, most of their information is accessible. It makes my job super easy because I could access all of their reports and get that data and show it to the public. New York Power Authority, about 2/3 of their information is also openly accessible, which is great for me. And New York Power Authority and then Ontario Power Generation, I haven't found any reports that I can access online. So when we look at some of the data that's not accessible to the public, so this is a report from New York Power Authority that it's not accessible to freely download, but I was able to do a data request, and they did send the report to me. So this is tail race surveys, and that's basically where those photos that Stephanie took, that Abe talked about, these eels are found in tailwaters of the dam. So these are eels that have gone through the turbines and have been injured or killed by the turbines. So the New York Power Authority, they do a survey every year for a few weeks when eels are migrating through those turbines. So when eels try to get back to the Sargasso Sea to spawn, they have to go through these turbines. And this is information that, it wasn't accessible to the public, but through the Great River Report, we were able to get the information, crunch it, and make it accessible. So it just shows the trend that mirrors that lovely eel ladder trend that Siena showed. But basically, you can see that the number of adult out-migrating eels have decreased through time. So you can see here, this is 2000. Up to 2025, and we have less eels that are migrating through the turbines. And this is basically the condition of the eels in the survey. So we have severed and alive and decomposed, and so they're going and they're counting these eels every year. This is data that is accessible to everyone, but I had to sort of do some calculations to make it, to pull out these numbers. So this is a paper that has data from 1997. So it is a bit outdated, but these proportions still stand true today. So this is silver eel escapement. So it's the adult eels that are migrating back to the sea and ready to reproduce. And if we look in the upper St. Lawrence River, 48% of those eels don't make it to the sea. So they suffer mortality mainly through hydroelectric turbines. So that's the number of eels in red here. That's sort of like a percentage of eels that don't survive going through those hydroelectric turbines at both the dams, so the Moses Sounders and the Beauharnois. We actually have very few eels, silver eels, so those are the adult eels that are captured in the fishery. So just to show you the comparison between the fishing mortality and the turbine mortality, and then we have 52% that do make it back. So getting that data, I am able to mobilize it and share it with the public to hopefully empower community. And I'm going to pass it back to Abe.
Yeah, and I think in these circumstances, this data allows us to act and start organizing. And I think that's that organizing that has allowed for this conversation to keep moving forward in a lot of ways. In particular, holding DFO accountable. It's really beautiful to see how so many different peoples and communities are coming together to organize, and not just depending on DFO or OPG to release this information, to organize as communities and use our voices, that this is not okay. And this is what this looks like. We hosted a eel symposium in Akwesasne, where we brought together First Nations with the DFO and the network to have these conversations and try to identify some future goals. Me and Marianne attempted to meet with an MP, but we met with their staff, and it was just really fun to learn about this really interesting dimension of the Sarah and the issues with it. I have had the lovely support and work of trying to organize in Akwesasne and sort of leverage some of the different environmental work that's already happening and figuring out how to tap into that, because everybody is busy but deserves to have access to this information in a way that is kind and gentle. And then we have the eel art competition that is happening, but I want to honor the eel gathering that happened through Leora Gansworth down in, I want to say, Kingston, New York. And we went to the Delaware River, where I got asked a really important question. When was the last time I saw a live eel? And I had to reach really deep for that, because I grew up seeing them dead on the side of the river, decomposing. And this sort of comes back again to that critical piece. My mentor, Carol Geha, who passed away in 2018, and just offered up this really beautiful teaching to me to think about the life of the eel and what they represent and how they overcome barriers and challenges to their existence, but at the same time need our support and our help. And so that's us, and this is sort of this idea of hope, that reinvesting in community and this recognition of this connection that things have changed, and some of these ideas that we've been sort of perceived within policy of incremental change. These policies can change. We see America, we see Canada, okay? Bill C-5 was passed in 2 weeks. We as First Nations were given barely any time to review it. And so there is opportunity to create change. We've been lied to. And so the hope is to reinvest into community, to use our voices and get out there, and hopefully that we can save the eel. And if that comes with some shaming of some governmental agencies and some industry, so be it. This is what I'm confident in and I'm believing in. Tuknigo.
Thank you so much. Thank you. I think we don't have any time for questions, and we do want to announce the winners for the eel art contest. Zach, do you have those slides? So yeah, come and find Abe and I if you have some questions. Sorry for the short timing, but so the River Rapport ran an eel art competition, and we got over 60 submissions of gorgeous art, and so we want to announce the winners. Do we have the slides? Yes? Okay, so we'll just wait a little bit, but so we got there we go. Okay, so I'm going to pass it over to Abe to announce the winners.
Alrighty, drum roll everybody. And so these are our gratefulness to the judges. I messed that one up. And so our winners are The Cutest Eel by Charlotte Emery. Come on, look at that eyes. Eels really do have eyes that look at you like this. I'm like, this is amazing. Most abstract. Ooh, James Magahi. Most unique, Layla Farmer. Spookiest eel, yes. Sovereign Benedict, yes. Most scientifically accurate eel, Mila Benedict. The happiest eel, Mavi Benedict. The best technique, Emma Andrews. Runner up, we have The Most Intricate Eel by Grace Soares. I almost thought that was Vicky Ransom for a second. And Outstanding Youth Submission, Sawyer Fine. And then runner up, second place, Simon Garnio. Am I saying that right? Simon Garnio? Garnio. Okay. And outstanding adult, Yagut Niguliosta. Yes, we all seen it out there.
This is a beautiful quilt that you see it in the front. It's drop dead gorgeous. Yeah, so we'd like to present the award.
I just wanted to let you know that it wasn't easy to do. And the reason it wasn't easy to do is because I got to look at some of the photographs that Stephanie Hildebrand had taken, and a lot of them were eels that were dead or dying, and it was like very hard to look at. And she warned me before I looked at it that it would be hard to look at. As he said, we've seen a lot of dead eels in Akwesasne, and so I was all excited when I saw the live eels that are on the quilt, and I thought, wow, these are beautiful eels. Then I saw the photographs, and I thought, how can we get people to look at eels that have gone through the turbines without them just dismissing it? So those eels that are in the center, one of the things is to bring the baby eels in from the ocean, and then have the nice mature live eels swimming towards the center, and then have them focus on those eels that went through the turbines. The pictures I saw, there were hundreds of eels that were dead in the bottom, and it was like my kids were kind of grossed out. I mean, everybody was. So it was like, how do we take that picture and get people to look at it and maybe not know what they're looking at, but as soon as they realize it, to be affected by it. Affected by the way that I was by Stephanie's photographs. And a lot of people are, I'll have to tell you that. While I was in the Adirondack Museum is when I was working on. So many people had so many stories about the eels. I learned so much just sitting there cutting out things and sewing it, that men, women wanted to tell me all of their eel stories. So I know people care. I know we can change things. And I went to the gathering that we had down in Kingston, and I was pretty depressed to go there. But I learned a few things that really brought my spirit up. One was that Quebec City, they said, well, we have lots of eels up here. We have lots of eels up here, and we think that they'll survive. And so that was enlightening to me. And then one of the individuals just said, maybe we shouldn't let them go towards Akwesasne and towards Baharnwa, because if we don't, then they won't get caught in the turbines, right? And then it was AB said, we can't do that though, it's their natural path, right? So it's like, we have to be careful of how we mess with nature in order to save these eels. But one of the things that also made me feel better was that technology is changing. We have an art exhibit in Canton, and you should all go see it. There's 10 artists from Akwesasne are showing their relationship with the environment. And the reason we did it there is because that's supposed to be a seat of knowledge, right? Canton is supposed to be Clarkson, St. Lawrence University, SUNY Canton. There's a lot of smart people there that could figure out how to fix this thing. But when I was in Kingston, they told me they do have a solution to this. And this is the inclined plane that's screwed so that it would be a turbine and it would raise the eels instead of cutting them up. So I just want to have the chance to be able to tell OPG to screw itself.
So we do have some other winners. So this is the outstanding adult submission. And it was also the runner-up, second place for the most creative. And this was the winner for the most creative. So it's a beautiful beaded eel. It's gorgeous. And Fred Schuler won this runner-up. He was the runner-up for the most thought-provoking with this beautiful poem. And the winner, I don't think we have the winner up here, but the winner for the most thought-provoking was Jessica Farmer. Thank you to all our artists. And we will now take a short break, maybe like 5 minutes, and we're going to come back for the eel art competition. Or sorry, eel art workshop. I'm so sorry.
All right, we're going to get started with our workshops now. I know we have a lot to discuss, but we also have lunch, so hopefully you can have some of those conversations then as well. So we have 2 workshops going on right now. We're going to have the eel art workshop that's going to start in this room, do a little foray outside, and then come back here. So if you're interested in the eel art workshop, you can just stay where you are. And then we also have Watersheds Canada is hosting a workshop as well that's going to be outside and kind of focus on an appreciation of our shorelines. So if you want to take part in that, you can head up to the giant map in the foyer there and meet Monica from Watersheds Canada, and she'll guide you through that workshop. So you can go ahead and kind of pick your workshop. Unfortunately, you can't do both, but you get to enjoy one and maybe talk to someone doing the other one and hear what they learned from that. So yeah, with that, we'll get started, and I'll pass it over to Kathleen Vaughn to kick us off for the eel art workshop.
So just give people a chance to sit down and think about art, do art. So I'm Kathleen Vaughn. I'm an artist and a professor of art education from Concordia University. I am a great lover of the St. Lawrence River and a huge fan of any activity that brings together different ways of understanding and knowing the river with a view to respecting our fellow creatures and providing safe ecosystems for us all. I'm here with 3 of my collaborators and colleagues from the university. We'll each introduce ourselves in turn. I told you a bit about me. I am part of, I lead a project that is funded by the Social Sciences and Humanities Research Council of Canada, called SURE for short, more on the social sciences side than the science side, sometimes it's the science world. And it's called Learning with the St. Lawrence. I've been doing that for 5 years and now have another 3 years worth of funding to think specifically with scientists in an art science exchange and to reach into all different kinds of communities to bring together different knowledge bases. I wanted to give you the opportunity to see what we saw. This is a picture that I took in August when those of us here visited the Moses Saunders Dam and saw a little bit of the ladder and the experience that the eels have. So this is just a picture from the eel ladder, the base of the eel ladder at the river where the eels start to climb in. On the bottom left, you have the model from the visitor center of how the eel ladder functions. And on the top, a living eel, yes, a live eel that has made it up through the ladder into the system and is going to be released into the waters above the dam. This particular grant has a collaboration with the St. Lawrence River Institute, a kind of a research center that I have followed and been connected to for many years. And it was proposed to us that we focus our activities on the American eel specifically. And so we have been learning a lot about the eel as non-scientists and are immensely moved by the stories that we hear. I had the great privilege of being one of the judges of the art contest, and I must say I had enormous hope looking at all those extraordinary works that people had created, thinking this is what people can do when they put their imaginations, their hearts, and their minds together to create something artistic. We need science, we need art, we need traditional knowledges, we need all kinds of things in order to try to make a difference in the world. I'm also encouraged by the words of children's novelist Catherine Rundell, who suggests that the imagination is the sharpest tool of ethics. And so hopefully the arts can allow us to sharpen our imaginations and apply that to our ethical understanding. I'll just tell you briefly what's going to happen. Because this is a research activity, believe it or not. We're figuring things out with you and hoping that you will participate with us in some aspect of the arts activity that appeals to you. We'll each talk a little bit about the different kinds of activities that will be on offer in this session. And then there will be a movement outdoors, as Erin was saying, so that we'll be able to be outside with some movement prompts. And then we'll come back inside and have the opportunity to do some art making, some stamping, some drawing, some stamping, different kinds of visual art. I'm particularly interested in art that engages many people, community-based art and things. This is from the symposium. The picture on the left is from the symposium in 2023, when I was here with this collaborative textile project. If you were here at that time, you might have seen us at the back of the room with people stitching, joining us to stitch during some of the talks that scientists were giving. We were creating an artwork that is a mural size sort of interpretation of some of the species of the St. Lawrence River using cyanotype, which is the blue photographic, the background blue like part of the river, eco-printing with some shoreline plants, which are some of the imprints you see in the middle, and then natural dyeing with plants from the shoreline and other local plants as well, which are all the different colors you see around the perimeter. So this is about 175 people worked on this over a 10-month period at different venues, including at the Shoreline Festival. And so that's some of the kind of work that I, as an artist and an educator, facilitate. But I'll pass it on now to my colleague, Shannon.
Hello. So I'm Shannon. I'm in the art education program with Kathleen. What do? I made coloring pages. That's what I did. So we visited the dam. We talked with people at the River Institute. And so I was just thinking, I didn't know anything about eels before. I didn't have any, I didn't think about eels. I probably didn't even really think that they know about them existing, to be honest. But then once I learned about them, I was just like amazed by them. And I started having these realizations that there were lots of things about eels, for example, that they're very cute. And so that was the first question I had for my first coloring page was, are eels cute? I wanted to ask people, what did they think? Are eels cute? The other one, you'll see them at the tables if you want to color them. I was also thinking the question, oh, sorry guys. Okay. I had 5 coloring pages. The second question is, are eels beautiful? I was really amazed by the glass eels and how beautiful they are underwater. Are eels brave? I was thinking about like their migration. They like go through the river, they go to the ocean, like it's kind of crazy. Are eels creative? They're very creative. They have all kinds of ways that they navigate and figure out new pathways. And the last question was, are eels important? So obviously we've all learned now that they're very important. And those are just the questions that I wanted to ask people and have people think about, especially people like me who don't really know much about eels. Yeah, I think that's kind of about it. I also, for the next slide, I also, Kathleen asked me to make some prints. So I'll be doing that in the lobby out here. If anyone wants to do some printmaking, I made some stamps, which will be up on this slide, about the eel migration. And yeah, I'd love to chat with you guys more about that.
Just to add, we're choosing art forms here that are accessible and easy to use. I mean, obviously, there's a whole range of different kinds of activities of art. Coloring pages are wonderful things that anybody of any ages can do. We hope you'll take some home or use them here, take them home, enjoy them with friends, family, loved ones. Printmaking has a huge history in activist engagements. You know, the people would make banners and signs to try to affect policy, to bring about change in some way, to share messages. And so what we have are small stamps outside up at the upper lobby. for you to try. Again, we'll set up the inks later on for you to try if you wish to, and just to have something to take home later if you would like that. But then,
Hello. It's a little bit tall for me. There we go. I'm Hannah Jacob. I'm a PhD student at Concordia University. I'm actually from Missoula, Montana, so you can go and tell the world that you met the original Hannah Montana. You'll always remember my name now. Here on the screen, I have a little bit of a taste of what we're going to be doing today if you join me for my section of the workshop. I am a scientific illustrator, so that is my practice. And I like to, I guess there's a bit of a colonial history to scientific illustration. It was very problematic and took advantage of a lot of species and peoples and their knowledge. So I like to work in a really careful way, in a really observational way, where you're getting closer to an animal in the most ethical way we can. So here today we'll be using photos, and we have the model of the American Eel River Institute to be using as inspiration. Scientific illustration, it's pretty hard to define it. It's kind of just a way to visualize scientific information through drawing. Typically, I use watercolor and pencil and add highlights using acrylic and whatnot. But today, we're going to be sticking with graphite, and I'll go into detail for those who choose to come to me. But I just want to give a little bit of a I want to make sure no one is afraid to draw. There's a lot of stigma around drawing, especially for adults, because we're told our whole childhoods that we're not good enough, or it can be kind of scary. So I just want to make sure no one's afraid and thinks that they can't draw, because I'll walk you through it step by step, and I'm sure whatever you leave with today, you'd be really proud of. So here is a finalized kind of scientific illustration depicting a species on the left that I worked with in Europe, actually. It's sea rocket. But what we'll be doing is more like the illustrations on the right. So taking notes and observing and kind of going in and looking at details of a species that you might not notice when you're just looking at it. So drawing really becomes this way of knowing and way of becoming closer with something in nature that you wouldn't get if you were just glancing at it. It really is a deeper process that requires a lot of attention, and it really sticks in your mind. And it's this way of embodiment that I think is really valuable. And so here are some of my past illustrations of salmon and an ecosystem. So trying to, on the right, the ecosystem is like a really complicated version of what you can do with scientific illustration. Thank you very much. I hope you come.
Hello, everyone. My name is Paulina, and I actually come from the shores of the Baltic Sea. That's where I was born and I grew up. I'm gonna talk a bit about my background. So I come from theater and performing arts background, and here are a couple of projects that I work with. So on the left, you can see Bread and Puppet Theater that maybe some of you know about. It's based in Vermont, and it's an activist theater working a lot with political activism, but ecological as well, as we all understand. It's very much intertwined. And this is an iconic puppet of Mother Earth that is operated by up to 100 puppeteers and volunteers, and it's crossing the whole playing field. It's a very physical work, and this is where embodiment also and movement comes in. Another project on the right is Acting for Climate, a performing arts company who has a branch actually in Montreal, but their main kind of base is in Europe. And it's a project I worked with. It's not me on the tree, and also both of the pictures are not mine. But I worked as an executive producer and tour manager with them. It was a performance promenade going through the forest, accompanied by storytelling, music, and circus performance. Next slide. So I also have not a very much linear path, though it depends on how you look at it. 1 year ago, I moved here to Canada, to Montreal, and to St. Lawrence River, with which I was trying to connect in different ways. I'm in an interdisciplinary program, mostly intersecting performing arts, performance studies, and environmental humanities. The lab I became part of because they had a group working specifically with waterways, exploring waterways through ethnographic research. Ethnography is a qualitative research method that looks at how individuals, societies, cultures, but also multi-species, different species and processes work. One of the projects I had the luck to work with this summer was ecosystem research creation by Geneviève Duperret, who spent about 7 years researching ecosystem of St. Lawrence. And ecosystem means industries, science projects, and ecosystem in more general understanding of this word. And she herself has background in circus arts. So she was working a lot with the movement and looking at movement and behavior within this ecosystems, and she created a show out of this. And on the bottom right, we're moving closer to the substance of the workshop that I will be giving today. Is a little movement workshop I gave on St. Lawrence. It's a low tide there. And it was in the end of a project when we were traveling between a few islands in the estuary of St. Lawrence, collecting data about plastic pollution on the shorelines of the islands. And I was collecting kind of ethnographic accounts. And in the end, I piled it up in a short, very short movement workshop, guiding my collaborators through the experience we went through a bit differently through our bodies and imagination. So I don't know if I just smoothly transition into, actually I can do this, wow. This makes me complicated. Whoops. Yeah, so what we plan to do is we're going to start with a little movement exercise outside. I really encourage you to come with me outside. It's going to be very refreshing after these 2 days being closed and sitting stiff. It's going to be a 10-15 minute, very simple movement prompts exercise. After that, we're gonna come back here. There's going to be a station with scientific drawing and printmaking outside. The workshop originates from my fascination with the movements of the eels. I myself, as well as Shannon, knew so little about eels just before we met with the River Institute. I got fascinated reading about them and listening to the River Institute staff members about their migratory route through the oceans and the fresh waters. I got fascinated by their shape-shifting, how they change throughout their life cycle. Start moving already. And I got fascinated by them as somatic beings, how they move, how they can climb the stones apparently, climb up waterfalls, how they can slither through across the land between water bodies. That was fascinating. And I was like, okay, so maybe a movement would be a good tool for me to explore it. So Western science often studies stuff through observation or experiments. Now I invite you to experiment a little bit with yourself and to think through movement. One important thing that I want to say that I don't want to think, I don't want you to mimic or pretend to be an eel or anything like this. I want you to think and be with the knowledge and mysteries, the absence of knowledge, which is a lot, of eels' movements, and I will guide you through this. So what I'm gonna do, I'm gonna give you very simple prompts, and we're gonna move together, and you can move as little or as much as you want, and you can also just observe and see what kind of weird stuff we're gonna be doing.
There's no right or wrong way to move. You can move just slightly, moving your finger, and that will be totally fine. But I will support you in this. What else should I say? Okay. Oh, that's it. Yes. Well, we're going to be outside. It's about 10 degrees, so probably take something warmer, but hopefully also you're going to get warmer with the movement. Again, it's going to be about 10-15 minutes, and then we come back. And in the end of the workshop, there's going to be a tiny gift that I want to share with you that I'm trying to fish you to go with me. I think that's it.
All right. Thank you. So let's suit up and see what happens outside. Bella's going to follow us with the mobile camera so that the people at home can follow along and be part of this in their own context, should they so choose. Then come without. Or, you know, if you want to stay in color, that's totally fine. You're doing a great job.
I think they're
Yeah, it's really beautiful. There are more here, more different models if you feel like it. Okay.
All right. Okay. Now we're just
Okay. Let's do it.
Okay, let's go.
Okay.
Outside main entrance and just cross towards the trees.
Let's wait for people outside the main entrance.
Okay. Yes. I have enough cards. I think we should just pass, like, skip that. Us, not take the place. Okay?
Yes.
Oh, isn't it good already?
Maybe even here, Paulina.
Like
Yeah.
on the stone? So
Yeah.
they'll-
Yeah, it looks like they're
That's a good spot.
Yeah.
Just come to me, that's okay for you to come on the grass. Does it work?
Yes.
Yeah, it works. Yeah, I kind of was trying to find a little bit hidden, so it's more cozy for us not to open to all the, but also to have the river with us while we move. First of all, I'm sorry. First of all, I want to thank you all so much for your courage, for coming here. I know it might be not a very usual thing to do on a symposium, so thank you so much. And again, there is no right or wrong way to move. So please feel free as much or as little as you want. I want to start with just bringing ourselves a little bit back in our bodies and yeah, breathing a bit. So let's take 3 big breaths. Breathe in through our nose and breathe out through our mouth. Let's do it all together. And let's breathe in and breathe out. And breathe in and breathe out. Breathe in and breathe out. If you feel like doing any stretching, it's a good moment now, because then I'm going to guide you a bit more specifically. Okay. I want to start with our hands. Let's warm them up a bit. It's quite cold. And let's rub them against each other and rub them vigorously so you feel the burn between your palms. Okay, nice. And now you can do the washing kind of movement, like you're washing your hands. And be gentle to your hands. Take care of them. Nice. And now let's focus on our fingertips. Feel them. They're quite strong. There's so many neural endings there. Wow, they're so sensitive. And now let's try them on one of our palms. They're like peas falling on your palm. And then another one. I wonder if we can pass it around. Maybe if I can pass it to you and you can pass it on. And we can actually start it somewhere else. Anyone wants to start there and pass it on? Amazing. I will pass you more. There's more. There's such an abundance here. Yeah, fantastic. Amazing. Okay. And now let's see if you feel comfortable. You can tap on your face a little bit, on your head, wherever you feel comfortable, actually, all over your body. Tap a bit. Oh, they're getting a bit tired now. Okay, wonderful. And now I want you to imagine that you have pigment or paint on your fingers, and there's a 3-dimensional canvas all around you. So try and paint as many dots as you can all around you. And remember, there's space behind and up there and on the bottom. Wow, it's nice when you can paint so easily, no? Keep doing what you want to do. You can come back to the simple movement that we did in the beginning. Just explore between these fingertips exercises. So the eels spawn in the Sargasso Sea. There's a carpet of sargassum weed, and one eel releases up to 20 million tiny eggs. And they spawn there because it's warm and calm. That's where they get born. So now I want to concentrate a bit on our palms. So we just worked with our fingertips, and now we go all the way to our palm. How can it move in the space? It's quite soft. There's so many tiny bones in your palm. Try with one and then try with another. Look at it, observe it. Now imagine that your palm is like a little tissue. It's very light tissue, and it floats in the air. Maybe you can help it. You can blow at it, and it's going to go up and then falls down. Wow, the air, actually the wind is helping us now. You can try and catch the air. Okay, nice. And now both of the palms are active and they can interact between each other. I actually would want you to break out of the circle a little bit now. Just walk a bit in space. Sometimes it helps for your exploration. And what if this palm is leading you? It's not you who's leading you, but your palm is leading you around. And you can also interact between each other. Maybe some of them meet and pass around. Just say hello, you know, the palms. Not you, but the palms. They are the drivers. Fantastic. Just keep doing it. So little eel eggs, they transform into larvae or leptocephali. They're ribbon-like transparent creatures. Some compare it to a willow leaf. And they travel for thousands of kilometers, drifting with the Gulf Stream
towards the coastal shelf. Amazing, you're doing great. Fantastic. Okay, let's go further. What if we look at our arms starting from an elbow? Let's start from this part. But it already embodies your active fingers and the palm. Yeah, there's this big bone that's not flexing, right? But imagine that there is no bone and it's soft. Wow, boneless arm. And now the second one as well. Oh, one important thing I forgot to tell you in the beginning. The main thing I want to take you out of this workshop, I want you to enjoy it. Okay, now two, and they can also interact with each other. Okay. Coming to the coastal shelf, the eels get a little bit of pigment, they become yellowish. Wow. Now they can slither maybe. Oh my God, did I forget? I did forget. We're gonna come a little bit back. We rushed a bit. Let's go back to our fingers. We did forget one stage, but that's okay. We're exploring, you know, it's a kind of an imaginary trip that we're having today. Let's look at our fingers and see how far these bones are going. They're actually much longer than what we see as a finger. Okay, and now I'm gonna invite you to paint again. Let's imagine that it's very dark and we're drawing with fluorescent paints in the air with our fingers. What kind of figures can you draw? Can you move them very chaotically and then very synchronized? Oh, fingers are so strange. This is a glass seal stage that I forgot, huh? That's the one when they passed the Newfoundland and they're transparent but more eel-like now. And you can see a tiny, tiny eel there. And they can now climb across the stones and rocks. It's a beautiful scene to watch. Okay, let's go a little bit through this stage that we already explored a little, the arm, the boneless arm. These are the juvenile eels, the elvers. Okay, now when we're warmed up, let's go the full arm starting from the shoulder. And remember, they're all the dimensions around you. Okay, and activate it. So the fingers that are active, there's palm that is active, there's arm from the elbow. Really go with it and keep moving in space if you feel like it. I don't need to be in the center of this. We keep going further. And energy, more energy, so that you almost get tired. Almost your muscles start burning. Maybe think of a flag that's attached to a pole and it's flapping on the wind. How would that look? Yes, yes. But all the way from the elbow. Wow. And the second one, yeah, you're doing it. Fantastic. Elvers transform into yellow eels. And these eels, they're super, super energetic. They go against the current. They're attracted by the bubbles. They can climb up the waterfalls. And they spend in this form, in this shape, 7 to 12, maybe more years. That's how they travel through the fresh water. All right, maybe you can be guessing, but now we're activating both of our arms, but they're connected with one invisible string. And they're heavy. It's not so easy to keep both of your arms together. And it moves very slowly. Think of yourself after a beautiful big community dinner that you had with your friends. And now you're going home content, but full and slow and heavier than you were before. Oh, wow. Yeah, and all the energy is in your hands. But it's much slower, much harder to move, but with a lot of joy still. So the yellow eels transform into mature silver eels. They become larger, their eyes become 10 times bigger,
and turn blue to navigate in the dark waters of the ocean. And in this shape, they come back to the Sargasso Sea. And maybe we can come back to our circle. Maybe you can say hello to your friends with your fingertips. We completed the cycle. Thank you so much. So the eels are commonly known by indigenous communities as messengers. They bring the stories of the ocean, the news, the knowledge to the freshwaters. And then after spending years and years in the freshwaters, they bring the knowledge back to the ocean. And following this prompt, I prepared little gifts for you. I honestly did not expect that so many of you will be so courageous to meet me here. So if they're not enough, I'm gonna make more. And please see me and I'll give you to those who didn't get one. So I want you to pass them around in our circular current of the Sargasso Sea. And that's a little message that these are tiny different little messages that I received from different writing that I was engaging with about eels. It's mostly drawn from environmental educators, indigenous scholars, and artists and marine biologists. And yeah, thank you so much for passing it all around. Yeah, now you know all the tricks. Okay, almost, almost. Okay, well, the last messages are getting to those who were lucky to receive them, but I promise you that I will make the quick ones for you to get. I want to thank you again for joining me on this journey, and I want to give a round of applause to each other and to the river. Let's do this together. Thank you very much. And I think we're going to continue with the printmaking and scientific drawing inside. Thank you again.
And we'll continue into lunch if you wish to. We'll stay around for you to continue into lunch if you wish to, but obviously break off and eat whenever that makes sense. Thank you so much.
Okay. Around 10. I'm going to work on this. Thank you so much. Thank you. I'll look so I was diving on those are cool. Yes, I might. Do you mind if we stay? Yes. I know you're.
like make sense. And then we're gonna reach into color if you want. I didn't know what like, but you're like I would recommend like something feeling good about what you have with this, and then exploring something just trying to erasers and everything. And then you can just explore with colors like this. I know we're doing scientific observation, but if you want to make now if you want to go more creative, if you want to make some more color, so welcome to do so. Really, this is just all suggestions and say whatever way you want that. But if you want to look at the materials and the images and feel free to do. So when you're doing it's just all this is eels very unique in terms of like a species that
Well, I have some crayons.
okay, so we have some they can like work into different positions, right? So they don't always just look like a like they can be
So is this your thing?
folded and like in nooks and crannies. So it could be interesting if you want it. Pieces of the environment, but typically what we start with is like thinking about what's underneath their skeleton. So like I could even think about their skull. I could be like, and yeah, you can start whatever you want. So I would maybe start with like kind of like what I think the skeleton looks like. I don't have a skeleton picture for you. Using the vibe.
You know what?
Like just think about what that like
joining the art.
and then think about the line of their body.
Montreal has a very good.
How that would work in the position and you can kind of look at passing around to kind of figure out how you would want to show your eel. And if you're like, I don't want to draw full eel, I just want to focus in on its face, the eye, and its mouth and nostril. That's fine. Wanting to just zoom in. And then after that, you can kind of build out like the flesh and think about, okay, how is this looking? Maybe you want to do the body first and then incorporate the big dorsal fin you see that like blends together with the tail fin and this one's this one's the anal, which one's ventral. I don't know about, but this is what's cool is that you can kind of see how it all comes together by drawing. It's like so I will be bouncing around for questions, but yeah,
I actually,
pretty much
what I do is
my
the basic
beginning spiel. But once you get going, the more questions will come up and just please ask me. Start later than you were. You are trying to make a big mark, and then you can just go in later once you have the general sheet with more there's space over here too. Can I show you the table? Yeah, you can start another. Bring the supplies and whatever you wish. Definitely. Yeah. Yep, I can do that right now.
Like looking in Photoshop.
Yeah.
Hi, for those of you online, right now in person, we are doing some scientific observational drawing of the American eel. And so everybody here decided, and we're brave enough to do so. So if during lunch, during your break, you want to take time, if you have a pencil and paper, it doesn't have to be a sophisticated like art supplies material. Just anything you have. If you want to pull up images of the American eel online, it could be a simple Google search, or using iNaturalist is awesome. Pulling together a bunch of different images and kind of positioning your eel however you would like. It's an exercise where you can really see deeply parts about the eel that you wouldn't have seen before, and you get to know it in this whole different way. So I recommend you do so. And if you have any questions, are there questions that come through or any communication? You could try to field them to me, but yeah, so I recommend you do so. Thank you.
Testing, testing. Hello, everybody. If we could all take our seats, welcome back. I hope you all had a nice lunch. I know I did. And welcome back to our final session of the day. This is session 8, Technological Advances for Monitoring and Understanding Ecosystems for the St. Lawrence River Watershed. So our first presentation for this session is Expanding the River Environment and Sensor Observation Network, codename REASON, to improve water quality monitoring capabilities in Akwesasne. And this is, please welcome to the stage, Logan, Lazore, and Erin Smith.
Great, thank you, Erin. All right, so yes, we've got a bit of a mouthful of a title, but yes, as Erin said, it's Expanding the River Environment and Sensor Observation Network, or the REASON Network, to improve water quality monitoring capabilities in Akwesasne. So first, what is the REASON Network? So it's something that was actually started by Michael Twiss when he was at Clarkson University. And so it was using these high-frequency sensors to monitor water quality in the St. Lawrence River, and has been used to bring kind of different types of findings about understanding the flow within the river and the movement of different particles. And so he's had these sensors set up in different locations, but today we're going to talk about the last few years where they've been monitoring at the dam, but also in Akwesasne. And so we'll be specifically talking about the data and the findings from having the sensor in Akwesasne.
Yeah, so I took over the monitoring from 2023 to just the beginning of this month. And I really enjoyed being a part of this project. It felt really nice to go into my own community and be able to conduct water quality testing. And kind of establish that data set, especially for a community that evolves a lot of their ceremonies and teachings around the water. And some of the other water monitoring or water quality testing that we did was this REASON project. We did boat sampling, and then we also used water rangers and YSI every time we go out for different projects in the community. And this, and yeah, while this project helped strengthen the relationship between MCA and River Institute, for me, it felt like I was able to give back to the water for everything that she's continued to provide for us, for our own livelihood, and putting in that effort to monitor her health was, for me, in my way of showing my gratitude, and hoping that she will continue to provide her support for our ceremonies, our teachings, for many generations after us.
So we have up here just a few of the project goals, overarching goals we had for this part of the project. So first, of course, collecting water quality data from these 2 monitoring stations. As I mentioned, today we're just going to focus on the one at Akwesasne. And then with that data, perform some analysis on that to understand some of the temporal trends within the river. And then from that, of course, to develop recommendations to guide planning and a long-term water quality management plan for Akwesasne. So this is really about building capacity, building data to help inform future water management.
Here's a good visual of what our setup looked like at the St. Regis treatment plant. Before, where I was standing, our setup used to be there, but we find that we kept getting in the way of the workers, so it took a bit of trying to figure out what worked best for them and what worked best for us. And it's a little bit hard to see, but the sensor is just behind that little wall there, and the outflow from the sensor was able to just go right back into a drain, so it kept everything nice and clean. And I think we can go, yeah. And this, so some of the challenges that we had during this project was, for some reason, St. Regis and SNI are really well known for their power outages.
it could be a beautiful day, and if there's one little gust of wind, the power will go out. I don't know why it happens. But so that's something that we were struggling with in the beginning, but we were able to just get like a power bank for the sensor. So that helped with that. And there was a lot of like connection issues with the laptop to the sensor. But a lot of times we were able to kind of troubleshoot that. It would just kind of make the monitoring or the data collection just take a little bit longer, which was fine. And then, yeah, we had a bit of priming issues and a little bit of wiper issues, but the intake was always like super clean, so we didn't have to worry about that a whole lot. And yeah, infrastructure was a little bit of a hiccup. The samples that we were collecting like came from this little like well, I guess. And like periodically it would go through like chlorinization, and we weren't really sure how that was going to impact our water samples. And that's whenever we started going out to do boat samples, so that we can actually get like the raw data samples. And while, yeah, there were some challenges, some of the successes from this project was that in the beginning, it was just me going out and doing the monitoring. So I would go every single Monday for however long I was doing it. But this year, I was able to teach our summer students the data collection process, the calibrations, the procedures for grabbing the water samples. So that really helped with staff capacity at the MCA Environment Program, which was nice. And then kind of showing them like the importance of why we do water quality, and then yeah, just teaching them like to give back to the natural beings that sustain for us, no matter like the treatment that we give back to them, and hoping that they'll continue to provide for us for many generations to come.
Okay. Oh, okay. So now I'm going to get into the data piece of this. So Logan talked a little bit about, yes, how MCA was able to adapt to these different issues that came up, and constantly kind of learning new things about these sensors, how to adjust to them. And then, you know, after Logan and her team went out and downloaded all of the data, you know, worked out all the hiccups, they handed off the data to me to work on to try to understand what are some of the trends that we're seeing with this data. And so up here, I've got a graph with the different parameters that we were measuring, and this is the raw data that was downloaded. So you can see, you know, there's a lot going on here. If you look at the top left graph, we have temperature here. And so you can see, we do see, even with the raw data, we see some seasonal trends, right? Makes sense. It went down in the winter. We had about freezing around 0 degrees in January, February, and of course went back up in the spring. So right away, that's a good sign that it looks like we're measuring conditions in the river. As Logan mentioned, right, of course, it was set up in the tank within the water treatment plant, but we also wanted to make sure that that was actually accurate to what was going on outside. So that was a good piece of evidence we have there for that. But you might also notice there's some weird kind of lines and gaps and stuff going on. And of course, you know, with any science project we do, it's never going to be perfect. There's going to be issues we'll encounter. And so I'll kind of take you through some of the things we've done to look at this data and still use it to get some useful insights on the water quality in the area. So with the raw data, there's a lot, right? Again, if you look at the top one here, this is just the raw data. It was taking samples every 2 minutes. So that's a lot of data over an entire year. And so it can be a little bit difficult to parse out the trends that are going on with that. And so one thing that I did was I started to average the data to reduce the data points that we had, and that started to give us a more clear picture of what was going on seasonally. And this is for chlorophyll A. So chlorophyll A is useful to tell us about algae in the water, right? How much algae do we have? We use it as a proxy for that. And so you can see, we started, this is kind of late summer last year. We had a early fall peak in chlorophyll A. So there's Again, potential evidence, maybe there was a bloom going on around this time. So right away, that gives us some key information about going forward, this would be a good time to go out to monitor for algae blooms in the river. Then we see a decline through the fall into the winter, which is as expected as we have lower temperatures, lower productivity in the system. And then an increase through the spring as well, again, as expected with the seasonal changes, increasing temperature, increasing nutrients going into the system. So already you can see with, this is 4-hour average data, and then daily average data as well. You can see these trends starting to emerge. As Logan mentioned, right, we wanted to make sure that what was being measured within the water treatment plant was reflective of natural conditions in the river, right? It's hard to show because we showed like what the inside of the treatment plant looks like, but that's connected through a pipe that does directly go out into the river. And so to check that, I was not there, I was sitting at a computer for all of this. Logan and some other members of the River Institute went out on a boat and took some sensors with them to check, is what's happening, what we're seeing inside the treatment plant matching what's going on out in the river. And so this data does confirm that. So at the top, we have data from the YSI that was on the boat, so that was basically being kind of dragged behind the boat to measure conditions over this period of about just under an hour. And then they also had another one of these sensors on the boat as well to, again, have that comparison between the different instruments as well to see if they line up. And so we can see, you know, roughly they follow a similar trend, different values, which is expected with different instruments, right? They're going to have a slightly different calibration, but overall we see similar trends for the two instruments on the boat. And then most importantly is that the bottom graph here, this treatment plant sensor, does follow a similar trend as well. And so that tells us that it does seem to be reflective. What's going on in the plant follows trends from the river. Another part of this project was the grab samples. As Logan mentioned, her team would, as well as downloading the data from the sensor, take samples, bring them back to the River Institute lab where they would be analyzed for some of the same metrics as the sensor, but also additional ones like total phosphorus, which can give some more insight into what's going on and what's maybe driving some of these trends as well. And so I did a comparison of an overlapping metric, which was chlorophyll A again. And so this is again looking at over the year, the trends that we see, right? So again, I already showed this graph. This is that chlorophyll A average so that we can kind of more clearly see the seasonal trend. And then we can compare that to our grab sample. And the grab sample one at the bottom, it's a little bit messy. We had different methods being used, but you can see that it does show some of the similar trends. But even in the spring when we get here, right, this peak that we're seeing in the spring with the sensor, we're not quite able to see that happening as clearly with the grab samples. And so that does show us the value of having a sensor like this, that it can provide more insight on what's going on in the river. And so for the project outcomes, again, is kind of reiterating some of the things that Logan talked about, but it increased water monitoring capacity at Akwesasne, again, with the training, the tools, learning about the setup, right? All of that was helping to increase capacity. We have this new information about seasonal trends that are going on in the river. And overall, it's just a strengthened partnership. It's another project that we're working on between the MCA Environment Program and the River Institute. So another opportunity is for us to work together and just learn more about the river together. Thank you.
Thank you, Logan and Erin. I think we have some time for a couple of questions. Are there any questions in the audience? I've seen a time's up a little sign there. Okay, well, thank you to Logan and Erin. In our next presentation, the River Institute will be looking to answer the age-old question, how many errands does it take to run a nutrient model? The answer is 2. So please join Erin Smith and myself for our presentation, Nutrient Mapping in the Lake St. Francis Watershed, How Artificial Intelligence Streamlined the Development of a Spectral Landscape Model.
Yeah, you can't get rid of me that easily, sorry. Yeah, so as Erin mentioned, we're going to talk about this SLAIN model that we've developed, and we'll try to go through fairly quickly. There's a lot of details, but we're going to focus a little bit more on how we used AI to improve this project and improve the workflow. So AI in research can be a bit of a controversial thing, right? There's a lot of different ways that we see AI kind of taking over different aspects of our lives. And within research, right, there's different components, different stages that it could potentially be used at, right? From coming up with project ideas to potentially writing either grant applications or papers, right? You get a little bit more kind of sketchy when you get to that idea. But another idea is how can we use it to improve our workflows, right? And specifically when we're doing things like coding, how can AI actually help us and how can we think critically about the ways that it can help us? And I see a few people laughing, enjoying our memes, so good. And so it can help us especially when we're thinking about complex problems. And so one of those complex problems is nutrients in our waterways. We've done a lot of work to reduce point source inputs of nutrients. So point source, you can think of things like outlets from like an industrial wastewater, right, or from sewage, right? You can see it, anything kind of coming from a pipe is a way that you can think of a point source. And we've done a lot of legislation that's improved our treatment of water so that we're having less of these. However, there's another source, and we call it non-point source, and that's more of a problem because non-point source is diffuse, so it's coming from a lot of different sources. It's difficult to track. It's not just there's that plant, it's coming from there. And so that is primarily going to be looking at things like agriculture. So when we get into more rural landscapes, we can have agriculture spread out, different types of agriculture, and so it can be difficult to pinpoint, you know, where are the nutrients coming from. And so one example of those types of landscapes is the Lake St. Francis watershed. So I think a lot of you are familiar with this. This is our land use map of the Lake St. Francis watershed. And so you can see, just I'll point out a few key spots. We've got Cornwall down here, that's where we are. And so that's the pink showing developed areas. And then as you kind of go to the east, we see a lot more agriculture in that yellow and orange there. And so that's kind of where we were thinking, we need to figure out where nutrients are coming from. From measuring water quality, we can see that there are high levels of nutrients in those tributaries. And so we wanted to figure out, okay, where are they getting into the tributaries? So kind of working backwards to the source so that then we can start to have management practices to reduce those inputs. And so there is a way that we can do this, and that's with this spectral landscape nutrient evaluation, or SLAIN model. Now this was previously developed by Mark McDougall, who previously worked at the River Institute, and he developed this for the Lake Simcoe watershed. So it's similar in a lot of ways to Lake St. Francis watershed. There's a lot of agriculture, it's spread out, and there's nutrient issues in the waterways. And so with this project, we were adapting that SLAIN model, taking the work that Mark had done and applying it to our watershed here.
Thanks, Erin. So Erin just explained what the SLAIN model does and how it works. So I'm going to tell you a little bit about how we actually make it and run it. And as well, both of us are going to go into some of the challenges in running this model and how we overcame them using AI. So the first part is to collect soil samples from across the watershed to identify where the nutrients are and in what concentrations. And then we also collect satellite imagery, which we use to process to derive some information from, and we use those to feed into the model along with the soil sample data to build a predictive model for where the nutrient concentrations are across the watershed. And then we have to test this model throughout using different seasons, and then we can share the findings with the public. So the first part is collecting these soil samples, and 300 sample sites were randomly selected across the Lake St. Francis watershed. There were 2 seasons of collection, both in fall and in spring, and these were mostly collected on public lands, think like roadside ditches, and I believe a few landowners also permitted the field scientists to collect samples on their farms as well. And the soil was measured for phosphorus concentration, and like I mentioned, this data was used to train this predictive model alongside that satellite imagery of the watershed. And this is something called ground truthing, which sounds kind of esoteric and mystical, but really all it means is comparing a model to real data. So the next step is collecting satellite imagery, and this imagery was collected from the United States Geological Survey using Landsat 8, which is a type of satellite that takes publicly available images all over the world, and it's divided into these different spectral bands, and that's just these different parts of the light spectrum. Some of them are parts of the visible light, like blues, reds, greens, and some are invisible, like infrared, near infrared, as well as some temperature data as well. And so we selected imagery for dates, that is around the same time that the field samples were collected, as well as selecting images that were clear and not covered in clouds and that sort of thing. So this data needs to be used to create what we call indices that tell us about features of the landscape, so primarily things like vegetation growth. Vegetation growth is strongly linked to soil phosphorus levels, so we can use this kind of information to predict which areas have higher nutrient levels. But this requires us to process these raw satellite images that are obtained from the USGS, and a number of steps are used to standardize this imagery, like clipping it to the watershed and scaling it for mathematical calculations and selecting the best quality images. I won't bore you with the exact details of how that's done, but I'll explain a little bit more about it the next couple of slides. And this imagery is also combined with geospatial data like elevation maps, which is processed in a sort of similar way, and these resulting data is then exported from ArcGIS so that Erin can use it in R. So the overall process is fairly straightforward, if you can believe it, but there are a lot of challenges that come with kind of a complex series of steps, and also a series of methods that is developed from somebody else's work, from years ago. So the original process was fully manual in ArcGIS, which made it very difficult. You can see it on the left there, a sped up version of some of the parts of the original methods. And being this fully manual process, it was very difficult to make any sort of adjustments to it or to make changes. So this is just part of what needs to be done for one of those spectral bands I talked about, plus all those indices. Every time there's like a mistake in any of this processing, or if you want to make a change to update something, I have to do this all over again from the start for every one of those bands, and that's just a big pain. It's very inefficient. But software like ArcGIS Pro has very powerful automation tools, but for someone without a strong coding background, it's difficult to take advantage of these tools and all they have to offer. So artificial intelligence was used to help develop this process at ArcPy, which is Arc's version of Python, making it both interactive and easy to iterate on. So it's important that while AI has many caveats, it's not perfect, but using it as a programming coach and assistant makes approaching these new kind of workflows much faster and more accessible. So working with these AI chatbots like ChatGPT, I can feed it questions about this exact process that I'm working with and the results that I want to see. And it'll help give me code that I can try or methods that I can use to improve my process. I can give it snippets of the code that I have, and it can help me explain things or debug things. And it's a very interactive process. You do have to understand what's going on, but it does save you a lot of Googling. And like this comic here, like, I don't know if any of you work with coding, but there's a lot of it that's just looking through age-old forum posts for people who have problems that are sort of similar to yours, but not exactly. So this really helps you hone in on the exact kind of information you need. And there are some challenges associated. As I mentioned, these bots aren't perfect. They're a great teaching tool, and they can really accelerate your workflow and how quickly you can learn. But they do require like a lot of babysitting. You have to have a decent base knowledge of what you're doing, your tools and your methods to be able to make the most out of it. Sometimes it can just give you bad code that doesn't really work, or it's outdated, it uses old methods. And you need to be able to go in there and like identify what's going wrong and then feed it, ask for a very specific solution. It can, it can hallucinate data that's just like not real, so it often repeats itself. It can suggest inefficient methods, so you do need to be on top of it. Thankfully, it's programmed to be very kind to you, and it's always happy to take feedback.
All right, so once Erin did all of the ArcGIS or ArcPy work, he handed off to me all of those files that he created. And so that ended up being a data set with the nutrient soil data, and then the satellite imagery data and all of those indices that he had calculated. And so my job was to bring those together and create a model that we could use to predict what we expect soil phosphorus to be across the watershed. So I started with building the model, so bringing together that phosphorus and satellite data. We use that through a method called random forest. I won't get too much into that, but basically it's a forest, so it's made up of trees. So we have different trees that sort out the data and figure out what's the best way to sort this data into different categories based on what we know about the phosphorus levels and the overlapping satellite imagery. So it created all of these trees for me, averaged them together, and then brought me this final result as the random forest model. Once I had that model, that model was developed based on the fall data set. So we had soil sampling in the fall, which we had overlapping satellite imagery with, and then again in the spring. And so I built the model based on the fall data and tested it on the spring data to compare and see, does the prediction from the random forest model match up with what we measured from the actual soil samples. And we found that it was actually pretty effective with its predictions. We had some error, but overall, when we're looking at this map is those predictions from the model. And so you can see, right, the red are the hot spots or the higher nutrient levels that it's predicted across the watershed, and the green is the lower predicted phosphorus sites. So we can see, right, that in its kind of eastern portion of the watershed, there's some higher levels there, and also in the northeastern up here. So that gives us an idea of where should we be focusing those management priorities. And so with building the model, similar to Erin, I was encountering some issues. And so I took inspiration from him when he was saying, oh, AI is actually helping me with developing the code, and I kept getting stuck as well. And so I figured, you know, why not, other than, you know, instead of searching through all these forums like he mentioned, I can use AI to pull all of that data together and give me some insights on what to do with my code to improve it. There were some issues because this code was a few years old. It was using sometimes outdated packages. It wasn't always the most efficient. And so here's an example of one of the questions that I was asking ChatGPT when I was looking to check the accuracy of the predicted random forest model. And so you can see here, it gives me two situations to choose from, right? And as Erin's mentioning, kind of having it as a coach in a way is helpful, but you have to have that background knowledge to understand what route do I need to go with this, right? What kind of data do I have? So it's really important that to remember that it's a tool that we use, but we still need to use, you know, our critical thinking, have that background understanding. You know, I still have to read up and understand random forest models and how they work and what other people have done, but it can be helpful for some of the more technical pieces as well. And so, for example, it took this chunk of code, which actually continued longer, and turned it into this. And this actually has a few more functions added into it as well, right? So even with that, it's able to make a cleaner code, which is useful for me, saves me time, and then I can share this as well, and other people can use it more easily in different areas. And so with that, once we had all of our data, we brought that model together. An important part of this project as well was sharing the findings, right? It's not useful for us to create this model and just hold it for ourselves and be proud of ourselves for making a beautiful model. We need to share it with people, and especially with people that can actually do something with that information. So we partnered with the RRCA and Alice to go to some of their events. Here's Erin out in a farm field sharing about the SLAIN model and what we did. We also created a story map that's live online for people to explore, learn about nutrients, and learn about what we did. So those are some of the ways that we shared that information. And going forward, we're planning to do more with that as well. There's a second iteration of the model that we want to build, considering how the nutrients are going to run off into our waterways. We want to add some drone components to get some fine scale imagery as well that can, again, improve our model. And then we want to integrate this with best management practices. So again, we're working with RRCA and Alice and OMAFA now potentially to work on the next phase of this and how we can use it to prioritize best management practices in the area. Thank you.
So I think we went pretty over time there, but we might have time for like one question, potentially, if there's anything burning. Yeah, come over.
You were asking about nutritional runoff or whatever it is, but the nutrition, doesn't it actually help with the river's health?
No, so for the nutrients that we're looking at, it's a complex question because in the main stem of the river, yes, nutrients, I'm talking about phosphorus and nitrogen, they tend to be relatively low, but that's mostly influenced by Lake Ontario waters. The near shore zone, which is where we use the water a lot, where a lot of biodiversity is, some of those areas are really high in nutrient levels. And so, and same with the tributaries as well, they can be really high in nutrient levels. So if you ever see, scums of algae on the surface, that sort of thing, those happen usually like late summer into the fall. Those can be detrimental to aquatic life and also to our ability to recreate in the waterways. So we do want to reduce the inputs into the waterways.
Okay, maybe one more question.
So just to confirm, the AI you used was ChatGPT. What model of ChatGPT?
So I can answer for myself. I was using, when I started on the project, it was ChatGPT 4.0 was the one that was out, and then while I was working on it, ChatGPT 5 released, and so I switched to that, and it was actually a lot better. Erin, do you have anything to add to that?
I think it would have been 4.0. I'm not entirely sure.
Okay, I think that's all we have time for, but Erin and I are here all day, so if you have anything else to ask us about it, we'd be happy to. So please welcome to the stage for our next presentation, Bailey Ruest, Past, Present, and Future Changes in Chytrid Detection around the Frontenac region and beyond.
And we're golden. Can you guys hear me just fine if I just talk a little more of a walkie-talkie kind of person? So this works out better. Hi, everybody. My name is Bailey Ruest. I'm in the Lougheed lab as well as the Wong lab, and I'm going to present to you the findings that we've done with chytrid fungus in the Frontenac region, and we're going to be looking at how that data can be helpful for future work throughout the St. Lawrence River. So before we begin, where's the clicker? There's the clicker. There we go. Aha. Perfect. So what is chytrid? So chytrid is just a kind of a short name for bee dendropatitis. This is a fungal parasite. So we know a lot of parasites. We usually think of worms and stuff like that. Fungi can also be parasites. This guy specifically infects frogs and salamanders. This one specifically is more impact towards the frogs, but salamanders can kind of act like a passive carrier. The optimal temperature for this guy to grow is around 18 to 25 degrees Celsius. So it doesn't like too cold water. It kind of likes water a little bit more on the warmer end of things. This fungus is actually a major component to the damage to amphibian populations around the world. So we theorize that about 500 amphibian species have been affected by this fungus. This impact is actually way bigger than the impact that things like cats and rodents have had on frog populations around the world. About 90 of these 500 are actually extinct in the wild now. So we can't even see them in the wild. They're only in conservation. And I think, what was it? 124 are facing 90% reduction in their population. So it's not good. It's not a good thing to have around, and it is currently present in a lot of areas. Another weird little thing about this fungus is that it's actually a very ancient fungus. It is like at the base of the fungal tree of life. And one of the cool things about that being so ancient is that they have this little cool thing that you're watching on the side here. This is something called a zoospore. So what is a zoospore? So when we talk about the way that they reproduce, they actually have something called a motile spore. So kind of similar to how our sperms move around. These guys are actually able to swim throughout the water to attach themselves onto their next hosts, which are frogs. So how it essentially works is we have this diagram here. This isn't a laser pointer. It does. Cool. So it essentially gets onto the keratinized part of the skin, and it will enter the skin of the frog, and it'll form these little thalluses, which are basically reproducing pots of more spores that will be released. The big problem with this is that frogs breathe through their skin. So having a bunch of fungus in your skin is not the best thing to have. Essentially, it's going to completely mess up its osmoregulation. It has a lot of difficulty being able to have their ions regulated. So more oftentimes, the frog will die of cardiac arrest due to this invasive fungus. Now, cool, it's invasive. But from where and when could we say this is from? So our current theories is that this invasive fungus comes from this little guy over here. Ooh, wrong button. Yeah, there we go. This guy over here, this is called Xenopus laevis. Ooh, there we go, close the mic. Xenopus laevis, which is the African clawed frog. So, if anybody's done biology, you might have heard about this guy. It is kind of a important species for a lot of our main research. It was also kind of big in the early times, like late 1800s, early 1900s, in the pet trade. And it also had a very interesting use, where it was actually a form of pregnancy test. So, I'm not going to go into detail about that. If you want to ask me about that later, go ahead. But it's a little fun fact about this frog. So this frog originates from Africa, and they believe that this fungi originates from that region. It's that or Asia. The facts aren't too cemented yet. But through that trade to North America, this is where we believe that this fungi was introduced. Thing is about BD. We're talking about BD when we talk about chytrid. There is another type of chytrid called B. sal. We don't have that in North America yet, but it is one that mostly, as I mentioned, this one affects frogs. B. sal would infect salamanders more often. So something to just keep your eye out. There are more species of chytrid that exist in the world. Now, that, we're going to talk about the other aspect of my research, which is eDNA. Now, if you're here this morning, you kind of got a good little earful of what eDNA is, but in case you're new, I'll go back through it really quickly with my little elevator pitch. So you know how they say dust is mostly human skin cells? So imagine being able to go to the corner of the room, pick up a dustpan of the dust, and be able to look at the DNA inside that dust and see every single person that's ever walked inside that room or walked through that room. It's kind of how eDNA works. With this, we're collecting free-floating DNA that's been shed off of many organisms. It allows insight in the species that are present in a location at a given time. The way that we've been collecting eDNA has been with a lot of work from our lovely Finn's team, as well as the Mohawk Council of Akwesasne. We've also been working with a group called SNAPS, which SNAPS stands for the Student Network for Amphibian Pathogen Surveillance. So they do a little bit more of a swab system for theirs, and I'll talk about that in a moment. Traditionally, when collecting eDNA, or traditionally when trying to find chytrid in any sample, the ways of doing it were a little bit more barbaric, you could say. The most common way of doing it was actually to take the amphibian sample and to trim a toe off of them, and doing a DNA extraction out of that to see if you find the chytrid in there. And before that, the way that they did it was a little bit more histological. So have you ever seen those like microscope slides of like skin layers? That's essentially what they did with the frogs. They removed skin layer and were able to look through the microscope to see is any of those thalluses present. Again, it's not the safest thing to do. It does harm the amphibians slightly. I mean, toe cutting's not too much, but nonetheless, you just want to be careful. So eDNA is a lot less invasive. The way that we do it is one of two ways. Finn's usually focuses on specifically the bottle collection, wherein you collect 1 liter sample of the water, you filter out, and you extract the DNA from that sample. As you can see, this handsome person over here in the water doing over there. The other one that you can do is swab collection. Pretty straightforward. You grab a frog, grab a swab the frog. And then you extract the DNA from that and you use that for to see if there's any chytrid inside of that. Yeah. So with all this in mind, there are different people who have done a lot of research on this, and the first one I'm going to talk about is the work done by one of our previous undergrad students, Mathieu Châtelain, and he did work in 2024 in the Frontenac region specifically. So that and also Kingston and Toronto. But you see in this figure over here, you see a bunch of either orange or blue dots. The orange dots are positive findings, and the blue dots are negative findings. So with that, you can see that in a lot of these city centers, there are a lot of positive centers and as well as some negative ones. So it's kind of not the biggest amount present, but you're still getting a positive detection with the eDNA detection. But one of the little weird ones we have over here is in the Frontenac region, you have this little pocket of plenty of positives. So if you do a little zoom in on that, That is the Queen's University Biological Research Station. So, what's going on there? This is one of the more interesting things where, yes, chytrid is being introduced to the area and being spread around due to the changes in climate, but how is it getting from point A to point B? Research equipment is one of the biggest proponents we think is the reason why it's being passed from region to region. CUBE's having a lot of students there doing research, we believe is one of the sources of this transmission of this fungi. So, not the coolest thing to find, but hey, it's good to know. Whenever we do all this stuff, we're doing something called qPCR to analyze the samples. To give you just the quickest little explanation of what this is, we're essentially looking for the DNA sequence of the species of interest that we're looking for, and we're replicating it a bunch so that we can see it a lot more clearly. And when it does that, it's able to cause a little fluorescent flash in our sample, and we're able to use the brightness of that fluorescent flash to get a quantitative amount of roughly how much in the original sample there was of that DNA sequence. Make sense, more or less? Cool. With that, we've been doing this with both the collection of the water samples, but we're also doing that with the swab samples of the frogs, which we've been doing since, I believe, 2022, and we are continually doing this experiment and continuing to extract samples from that one. So that's where we get to the swabbing. In the sample here that you see, don't mind 2024. I think that year they just didn't go to the site where they had a frog, so they mostly just collected salamanders. So that's why it looks a little bizarre. But if you can look at the other years, positive connection are these blue parts, and the negatives are the red. So first year, 2022, not a lot of positive detection out of 40 amphibian swab. This is both swabbing salamanders and frogs. And then you come to 2023, there's a little bit more. And then the work that I did this year, there's a whole bunch more. So with that detection, we're seeing that there's a increase in chytrid. Not yay, but you know, hey, found some data. So with all that in mind, we know that this fungus isn't good, as I mentioned, is a major impact to the frog population. Where are there a lot of frogs? If anybody has a nice little more watershed area on the side of St. Lawrence River, you know that there are plenty of frogs. Ergo, there's probably some chytrid in St. Lawrence River, which people don't really know because a lot of people don't do a lot of surveys in flowing water bodies because it's very difficult to detect anything in there with eDNA as we're doing with fins. But speaking of fins, with fins, we have collected eDNA samples from, I believe it started technically 2019, but we officially started in 2020. And we have samples collected from there all the way to 2025, from the Bay of Quinte all the way to Montreal area. So with that, we have 5 years worth of samples all along the St. Lawrence River to be able to look at a timeline of how chytrid has passed along the St. Lawrence River over time, and if it's present, is there any changes in that? Is there more chytrid in areas that are more developed, or are there chytrid in areas that are less developed? And not just chytrid, honestly. We can also do this with a bunch of other invasive parasites. Some that are affecting eels is coming to mind, and other fish as well. But nonetheless, we have all these sample points that we can use to our advantage to do this. Now, with all that in mind, what can you do? As I mentioned, chytrid is mostly passed around due to equipment not being properly sterilized, going from point A to point B, making sure, so, you know, people oftentimes will take their boats, bring it to another lake, not think anything of it. However, that is one of the major sources of transmission for a lot of these microbial invasive species, or species that should not be introduced into certain water bodies. So, what you can do is make sure that whenever you're done with your boat or vessel or whatever, take out any organic debris that was in that water body. Properly empty any bilge that you have in there, or any water that's in your boat. And also make sure you dry equipment before you put it away. Also, sterilize your equipment, especially if you know you're in an area that might have any of these things. Make sure one of the best ways we know to do it is with bleach, and also letting it dry for 30 days before you put it into the next water body. So it's a lot, but it's to limit these amount of transmissions that could happen. And with that, I would like to thank everybody at the Lougheed Lab, and as well as the SNAPS team, thanking specifically Lenny Shirozi. St. Lawrence River Institute, I thank you very much for helping me collect these samples. I mean, I was also helping with collection, but nonetheless. And also I'd like to thank the Mahawak Council of Augustosne for also aiding in collection of these samples. And I thank you.
Any questions?
If we have time, of course. Oh, fantastic.
Hello. I was wondering, with a lot of these projects, there seems to be like similar sampling areas, similar like test ranges and times. I was wondering if there's ways to like scale up the testing practices so that multiple researchers could effectively have steady streams of the samples at all times.
Yeah, so you're saying like if we're having all these sample collections there, can we use it as like something to share those specific samples with a bunch of people kind of thing?
Yeah, because
it's hard to do. This is mostly something that has to do with like costs kind of thing. So the process of doing eDNA, the filters themselves are pretty expensive, and the process itself is pretty, I wouldn't say too labor intensive, but it does require some labor. So to try to make enough to be able to share amongst a lot of people. So when we do the fin stuff, we're only making like, what is it? 1 liter, 200 microliters about, I think at the end of the day, and we have to ration that out to as much as we can. So there could be ways of doing this a little bit more mass-wise, but again, you need a lot of money to be able to do this. I think the filters come up to like, is it $2 per filter or $20 per filter? $2 per filter. So each run is $2, and you have to do usually 5 runs per site. So yeah, pricey.
Thank you for your presentation. I just had a question regarding the slide where you had the count of the salamanders and frogs.
Let me know. This one? This one. Or
we're still seeing the acknowledgements.
Oh, no, God.
Yeah. So, yeah, the one with the red. Yeah. So I'm wondering about the distribution between the two animals, and does it matter because you're just looking at the parasite anyway?
Right, so I believe they tried to keep it as best they could to about 20 individuals per site, so 20 frogs, 20 salamanders, hence why we have about roughly 20 salamanders at this site. But again, it was more just of what they had collected at the time, so it could be like maybe 23 versus 17, but roughly always 40 was the end goal. Make sense? Cool.
So you mentioned in terms of the equipment letting it dry for 30 days. How long would the fungi actually last without a host, and would that affect your water sampling for eDNA at all?
So when it comes to water bodies, whenever we do eDNA, the expectation for any sample that we have in there is that it's been roughly probably out, I think DNA has a half-life in water depending on pH, sunlight, so on and so forth, about 3 days. So by that point, we assume that the cell is probably degrading at that point. The cell doesn't have that big of a half-life since its goal is to get from point A to point B, infect, and reproduce. So to that scale, I'm trying to remember exactly how you worded your question. We don't know, so you're saying if like, how long do we know that that sample has been in the water body per se, right? Like, do we know if it's a new collection or if it's something that's been there for like months on end, right? Yeah. Right. So whenever we find in the water body, we can at least assume that if we're finding positive, like I said, they don't live that long. So any frogs in the area probably do have chytrid. So that's where we have that positive detection with the water samples. When it comes to swabbing, we're pretty much doing direct dermal. So we can pretty much do the confirmation that it is probably that frog that has chytrid. So the water one is, again, mostly just an assumption of what is in the area. But if you are detecting it, chances are it's present. Make sense? Coolio.
Thanks, Bailey. That's all the time we have for questions, but as I think we can all tell, the man loves talking about chytrid, so he'll be around.
It's just talking. I just love talking, honestly.
Okay, for our final presentation of the session and for the symposium, I believe, please welcome Alan Tian for his presentation, Developing an eDNA Workshop, What We've Learned in 4 Years of Capacity Building in Eastern Ontario.
Thank you for sticking around this long. I didn't expect as many people would be left in this room. So I'm just going to jump straight into it. I don't need to tell this audience that we're currently in the middle of a biodiversity crisis and that freshwater species are more threatened than basically any other group. And I think we've shown this slide 3 times now. So as our plenary speaker Steve mentioned, the World Wildlife Fund's Living Planet Index shows us that the global populations of wildlife are in severe decline, with 73% of monitored populations suffering decline in the past 50 years. An important factor to note is that much of this decline is occurring in the global south, in areas such as Latin America, the Caribbean, Africa, Southeast Asia. These areas are disproportionately affected by climate change, And they might also lack the resources for the widespread monitoring that we need for effective conservation. There's also simultaneously a massive deficit in global species monitoring efforts. Although great effort has been made in species classification, with over 1 million species delineated by now, modeling estimates that there's at least 9 million species existing on our planet. Some groups, such as fungus and insects, are woefully undocumented, and many will go extinct before we ever classify them, let alone make any effort to conserve them. Emerging molecular techniques have been making a huge dent on this, with the global DNA barcoding project set to classify more species than traditional Linnaean classification by the end of this decade. So this brings me to the topic of my presentation, environmental DNA. So as organisms interact with their environment, we all release genetic material. For instance, much of the dust around us is just dried skin cells. The rest is microplastics. Yes, very grim. And by collecting environmental samples, such as air, water, or soil, and then extracting DNA from it, we can get a snapshot of what's present. We can use multi-species detection methods, such as metabarcoding, or single-species detection methods, such as quantitative PCR or digital PCR, to assess species presence, absence, or even potentially population with many caveats. The exact definition of what eDNA is very murky. It includes environmental samples that consist solely of free-floating and adsorbed DNA, passive aggregators of DNA such as spider webs, bee pollen, dust on leaves, soil samples with many microbial or fungal organisms, or even bulk samples from deliberate sampling efforts such as insect malaise traps. Regardless of your definition, though, eDNA allows us to survey species of interest with minimally invasive sampling, and often at lower cost and without significant naturalist knowledge, which is a key factor. To be a good birder, you need to have a lifetime of birding experience. Additionally, samples can be collected ad hoc and archived for future applications and assays, particularly as techniques develop. So in the past 3 talks on eDNA, all of those samples were collected by the FINS team. They were the same samples. We can do many different assays on them. Everything from monitoring fish to monitoring pathogens. And we still have some left. It's also suitable for long-term archiving. DNA that's been purified and stored in a -80 or freeze-dried is stable for many, many years. And some companies are actually talking about using DNA as a storage medium itself, because it's very information dense. However, there's many current issues in environmental DNA. First and foremost, in the sampling and extraction stage. So eDNA is a very young field. When it developed, we started with many different protocols for sampling and extraction. We got those protocols from on water quality monitoring and tissue extraction. So even today, we mostly use, if we buy a commercial kit, we use something called a Kaigen blood and tissue kit, which is meant for extracting tissue DNA. Comparative studies on these protocols are often very limited in scope. Metadata collection and reporting are not standardized. So what metadata is information that's collected along with the sample. This can include everything from the date to the water depth to the filter pore size used, and many papers don't report a lot of these things. This isn't the fault of the paper necessarily or the authors, but rather the time, energy, and money are pretty limited, and every bit of metadata costs time to collect. But regardless, things like pH, which many of you would consider to be extremely important, only 9.9% of papers actually reported that for eDNA, even though it's a direct factor in eDNA degradation. Finally, fee-for-service companies, which many communities now use for eDNA work, might outsource lab work and analysis. So a lot of fee-for-service consulting companies, they've started out in other forms of environmental monitoring, branched out into eDNA as it's become popular, but they only do the actual sampling, and then they outsource to another, an academic lab like the University of Laval. And this creates a disconnect with fieldwork, which makes troubleshooting very difficult. There's also many issues with environmental DNA labs. Even the best labs have many issues. As a very young field, very few PIs actually started their lab with the intent of doing eDNA. And so labs are often not specifically designed for environmental DNA and are multi-purpose. If you're working with massive numbers of polar bear poop samples, like Dr. Lougheed is, or many fish samples, like the FINS team is, you're going to have lots of tissue and lots of sources of contamination. Shared apparatus and freezer space are major contamination sources. Very few pretty much no people can afford a dedicated -80 freezer for eDNA specifically. The ideal eDNA lab, which currently we're hoping to build at the Queen's University Biological Station and at the St. Lawrence River Institute, has a unidirectional workflow where you go in, you decontaminate, you extract your samples, and then you make sure that sample extraction is far, far away from tissue and where you do your PCR. There's also lots of issues in eDNA training and standardization. Very few to no personnel are specifically trained on eDNA sampling and lab work protocols. When I started, the protocols were totally different. We've been making things up as we go along. Protocols lack standardization. The consulting companies that I mentioned before offering eDNA services can often lack capacity for QA/QC and troubleshooting, and metadata is often neglected during sampling. When you're out in the field trying to collect samples, you don't have time to make sure everything's perfect. However, significant progress has been made towards high-quality standardization. So in 2023, there was a conference called Pathways to Increase Standards and Competency of eDNA Surveys at the University of Guelph. And all of this led us to develop our eDNA workshop. So this was the first iteration of our eDNA workshop. We developed it at the Queen's University Biological Station. So we took one of the labs that was used for fish dissections and microscopy and other things, and we brought up many truckloads of our molecular lab equipment up, and we tried to run a workshop. So we developed the workshop as one of the goals of Insert Frontiers, SSRHRC Frontiers grant. We also wrote a 60-page manual to accompany the workshop because there was a lack of literature on environmental DNA, and it lasted 3 days. Participants came in on a Monday and then left on a Friday, but Monday and Friday were for logistics. And we tried to fill a gap in eDNA training. So we did a practicum-focused workshop that did a lot of in-field stuff that covered sampling and extraction and put it in context with the entire workflow. We also had guest lectures from Biorad, which is the company that made the qPCR machine that Bailey showed us. Dr. Bob Hanner, who was a leader in the 2023 Pisces conference from the previous slide, and a general luminary in the field of eDNA metadata and standardization, and Dr. Elizabeth Clare, who's a pioneer in aerial eDNA. So day 1 was on sampling and filtration. Day 2 was on extraction and qPCR and digital PCR, led by the Biorad company. And day 3 was on metabarcoding library prep and bioinformatics. We did a mixture of lectures and practicums, where a lecture would be followed by a practicum. And we tried to focus on practical skills and applications of eDNA. We faced a lot of challenges since we were developing everything de novo. We had to develop a curriculum. We had to avoid overlap with existing courses. So a lot of us had taken courses. Mostly they were online because the previous years were COVID years. And they were, a lot of them were more analysis or theoretical focused. We had to develop collaborations with the previously mentioned guest speakers, and then we had to set up a molecular lab at a field station, which was the most contaminated space you could imagine. We also developed the EDA manual, so you can scan the QR code if you want to take a look at it. So up to this date, there's only a single textbook on eDNA, and it's only around 150 pages long if you don't count the index, so we're approaching that. It covers a general overview of eDNA, single species assays, how to make and apply them, metabarcoding assays, how to design sampling schemes, the bioinformatics of metabarcoding workflow, and then more recently we added eRNA, loop-mediated isothermal applications, and a suite of protocols that we've been using. They're not the best protocols, but we know they work. And we've continuously developed this manual. Every single year we get together and then update it, and it reflects our existing knowledge base and lab techniques. And in the future, we hope to include things such as long-read sequencing metabarcoding, metagenomics, which is to look at the DNA in a sample rather than specific markers, and other techniques as they develop. So these were the reviews of our eDNA workshop. So every year at the end of the workshop, I sent out a survey just to gauge what the participants liked and didn't like, and they were very happy. So the bottom of the workshops, the bottom of this starts at 5. So on every parameter, we basically rated 9 or above. Participants wanted an extra day though. They wanted more content, more in bioinformatics, project planning, and other sampling types. So in 2023, 2024, and 2025, we took those suggestions and then we incorporated them into the course. In 2023, we extended the workshop to 4 days. We separated days on metabarcoding and library prep and bioinformatics. We streamlined the Biorad demo from both qPCR and ddPCR to just qPCR. ddPCR was too ambitious and we couldn't actually fit it in the time frame. An interesting to note was that our participant demographics actually shifted during the course of the workshop. We went from mostly students, mostly undergrads and junior grad students, to industry professionals, some government officials. In 2024, we added loop-mediated isothermal amplification, which is on the bottom. Okay, that could have gone worse. So this is a technique that's been used in COVID testing, and it's isothermal, which means that you don't have to shift the temperature in the way you do with PCR. All you have to do is raise it to 60 degrees, and then if it detects the target DNA, it changes color. So you could use it in the field, theoretically. And then in 2025, we added an optional day-long long-read sequencing workshop run by Oxford Nanopore. Oxford Nanopore's equipment is very portable sometimes, and their smallest sequencer, the MinION up there, is around the size of a credit card. And so far, we've taught around 60 participants. And again, they were all very happy. Everyone rated us around a 9 or higher for usefulness and how likely they would be to recommend the workshop. And we've continuously monitored how they received the guest speakers, lectures, field practicums, lab practicums, how they saw us, accommodations and food. And every single year we switched up the guest lecturers. So we had Chris Wilson from the MMR, and last year we had an expert on eRNA. So in the future, we hope to continue our eDNA workshop. We put in new grants. There's a new building currently being built at the Queen's University Biological Station. So this will be a state-of-the-art molecular lab, so much less contamination. We've purchased new equipment, new pipetters. We hope to expand our manual. And then hopefully with nanopore sequencing, we can actually do a library on site and get results that day. Potentially metagenomics, and then potential collaboration with Pisces, other labs, and hopefully continuously collaborating with the FINS team and the MCA. And if you have any questions or if you want to be added to our email list, please contact me.
Thanks so much, Alan. I've been told if I take more than one question, I will see great bodily harm. So,
okay, just a quick question. So you said it shifted from academic to a lot of industrial presence. Like, why? What would be driving that?
I think there's been greater adoption of eDNA across the consulting sphere. As well as among NGOs and government organizations. So with this greater interest, there was more demand for training from people that weren't students.
Thanks so much, Alan. That's all the time we have for today. He's also a really great drone photographer, so follow him on Instagram. Okay, so that's the end of our session and the symposium. So thank you all for sticking around for so long. And before I hand it off to Erin, I believe it's time to draw for the door prizes.
All right. Whoa. My bad. There are a lot less in here today, so you all have statistically higher chances of winning. Can I get a drum roll? The first winner is Casey. It does. The second winner is Monica Seidel. Seidel.
Okay, you can come on up and you get a $25 coupon for the Outdoor Learning School and Store, and they have so much amazing stuff, so you're very lucky. That would be very nice. Is Monica here? Okay, maybe we'll just have to send it to her. Okay, that concludes our draw. And now I'm going to pass it over to Abraham Francis to close us out. But after he's done, please stick around because we have the River Strategy AGM happening here at 2:30. So welcome, Abe.
Well, heck yeah, with the successful drum roll. You know, I had this really deep moment of reflection while I was at the First Nations Great Lakes Water Gathering the past 2 days, and reflecting on the fact that, you know, how important this gathering has been to me. You know, one of the first pieces that I wrote and published emerged from this place, which I think is pretty powerful for me to reflect on, you know, thinking about Akwesaslono relationships across time, and how that kind of helped me work through a really difficult time and grief. And I think collectively, and I think the theme of this year, Changing Waters, right, if I'm remembering right, Changing Waters, am I crazy? But really connects with that, because I've often heard a lot of, like, a lack of hope in regards to the work that we're doing to protect the environment, to gain deeper understandings. When we look at legislation that is put in place that impacts us, the cuts in funding, and the way that globally we are seeing, I wouldn't even know how to fully describe it, but other than just like sadness. And I think that this is such a really important place of transformation and innovation, you know, and I'm just grateful to be a part of that, you know, to look at all this eDNA work and to think about how beautiful it was to see a partnership between MCA, the River Institute, and Queen's University, and, you know, being able to re-articulate that proposal, you know. And I remember meeting with Stephen back in those days, and there was just a lot of lack of hope in that moment. And then, you know, through community, through collaboration, we made something that felt impossible possible. And I think that's something that I really want to sort of communicate and continue to reinforce in conversations, is that hope does exist out there. And I think that we need to tap into that and keep moving it forward, but with the reminder that, like, hope cannot go without action. If we just sit there and just hope for the best, nothing's going to happen. But if we put our bodies behind it, we believe in the relationships that we've built, then I think that we can create the transformative change in the world that we want. And I'm just going to continue to reinforce those messages and tap into that. And, you know, and a little bit more moment, you know, the other day I was listening to Mike Mitchell do the opening, and he said, Ayadawahnuni, Skana Ayadawahnuni. And I've been studying the Ohandagaliwadekwa, which sounds crazy to say, because the version that you guys get is pretty consistent. But the version I get to see is always changing. There's always a new version. There's always a new word added to it. There's a new dimension, and I love that about my language. And what he's added to it was Skana Ayadawahnuni, which means we're making peace. And I really was proud in that moment. One, I recognized Mohawk being spoken from, like, a traditional knowledge keeper, but also to really tap into that space of how much is embedded within this teaching. And you'll hear some of these words in there. So I just wanted to share a bit of that insight. And so with that, you know, we've come to the end of this gathering, per se. I mean, we still have our River Strategy meeting, which you guys should stick around and learn a bit about. But to extend my gratefulness to the planning committee that put in all these months. I'm grateful to the River Institute, grateful to everybody that funded this, and, you know, just grateful for the opportunity to continue to return and provide the opening and the closing. You know, you guys got a digital version of me the other day, but I'm grateful to be able to do this. So with that said, I am going to hop right into it and close out our council fire. Yung yukwa swada hung CEO Scott Nigaliwesa, Danchi Dunu Halado ne Sogaya Dizo Negadi o Handa Galiwadekwa, Unguano Headsto. Agwego aska ndi the wahwet nuni ne ungwani gula dano de tenwalado ne ungwet sua, ne agwego skana agahageji de wadawonye, ne ganto o hunjage de wandalo. Tunyuto hak ne gwani gula. So. Agwego aska ndi the wahwet nuni ne ungwani gula dano de tenwalado ne, yikin istahaji o hunjade. Ne eji sego dai gotskawahage ona huda sua dai ngwashne skana ay dawa dun musage. Tonyoto hakni kwani gula. Agwego aska ndi dawa hwet nuni ne kwani gula dano de tinwalados ne onega sua. Jisego wados ay ngwahat dano watste dano askalago e ne wahetko skayo. Tonyoto hakni kwani gula. Agwego aska ndi the wahwet nuni ne kwani gula dano de tinwalados ne gajasua. Ne wahi kyun kiawis ne kaya dagan hatsala ne dosa ay dawadun kayake. Tunyito hak ni kwani gula. Agwego aska ndi the wahwet nuni ne kwani gula dano de tinwalados ne ohundet sua. Ne eji sego yon kiat dagenhas okyake gakwa sua dano okyake numpwa sua. Dawatsta. Tunyito hak ni kwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne onunkwa sua. Ne eji sego yon kiat dagenhas da yung washne yon ki junta. Tunyito hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne ohtela sua. Ne eji sego yon kiat dagenhas dano okyake da wanungwat salun yata ne da yung washne. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne gayentosala. Ne eji sego da yonat kawahage ne gakwa sua nona da wadun kayaks. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne gahi sua. Ne eji sego yatigos dano Kundo de buenio, ne guana, got ni hundesa, ne egisego ganalines, ne gahisua. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne ojinua sua. Ne egisego guadogo yodi odage, gina hoda yana dalihunde jihunjade. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne gundilio. Ne eji sego yon kiat dagenhas nona dawadun kayaks dana dawawistos. Tunyato hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne ogwele sua. Dano ne tiguana, wata jiniga guilodas, ne egisego tot nio de la hage, o na hoda sogonya heza o ne sungwaya dizo. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne ojidon agua, gisego yung guatunde ne galanios la di hawi. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne kisotagun ala diwelas. Ne eji sego azes anuni jiganagaluño. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne gaeli ne gawelage. Ne eji sego to nio de la hage ona hoda ne sogwaya dizo sagunya hezao. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano danchi dunu halado ne sungwachi akyo ke neka galakwa. Ne eji sego de hoswatedo jiyo hunjade. Tonyoto hak ni gwani gula. Agwego aska de wahwet nuni ne gwani gula dano de tinwalados ne yakisota asata neka galakwa. Ne eji sego yatigos galunhyage dano lowedeni lalagyes ne la diksa agua. Tonyoto hak ni gwani gula. Agwego aska de wapwet nuni ne guanigula dana de tinualados ne ojisto quasuma ne yago gyatsununya takwa ne yakisuta sataneka galakwa. Tonyata hakni guanigula. Agwego aska de wapwet nuni ne guanigula dana de tinualados ne gaeli nyungwedege ne e wahe luedeli hunde ayonkiat danona ne ungwet suma. Tonyata hakni guanigula. Agwego aska de wapwet nuni ne guanigula dano danchi dunu hala done sungwaya dizo ji agwego luena dao ne ohunjage jina hoda de sungwawi. Tonyata hakni guanigula. Ona wadali wan hodo nua wanizalade doga oteno sayungwatnigulha ize gine oze wada sundala ehkadi. Tonyata hakne swatnigula. So I just wanted to add to this with the closing here is this reminder that comes with it in just sort of another expression of community. If I forgot anything, there's this really important piece. If I forgot anything, if I forgot to acknowledge something or forgot to address something, then it is our responsibility as a community to carry that forward, to address it in our lives. And so within this sort of frame of thinking, I hope that what the messages we heard during this gathering, the knowledge we gained, that it empowers the people here to act and to make impact in the world around us. And you know, this gathering and so many different pieces of work I've been able to be a part of and to see come into being is just evidence that community works. And so, Tuknigo.
Great. Thank you, Abe. We really appreciate you giving us that wonderful closing for today. So yes, just a few technical things. So we are going to have the River Strategy AGM happening in this room, but they do need to make some changes to the room for the setup. So we will need everyone to exit the room fairly quickly so that we can make those changes and come back for the AGM. But I do want to just say a big thank you to everybody for coming and a thank you to our organizing committee as well for all their support and all the River Institute staff and volunteers that stepped up today and the days previously. So thank you.
much.